Hello there, and welcome to Life Beyond Seen, where curiosity settles in, the lights dim a little lower, and science takes its time unfolding. Tonight, we're travelling somewhere few humans will ever go, to a place so deep and so pressurised that even our strongest machines struggle to survive for long. It is a trench in the Pacific Ocean, nearly 7 miles down, where sunlight does not reach, where temperatures hover just above freezing, and where the weight of the water above presses down with a force that would flatten most living things in an instant. And yet, life is there. Not just bacteria, not just faint traces of survival clinging to rock. Entire creatures live there. Creatures with translucent bodies, creatures that glow, creatures with jaws that hinge open far wider than seems reasonable, creatures that look less like animals and more like unfinished sketches. If someone had drawn them for a science fiction film, you might gently suggest they tone it down a little. And yet these beings are not fiction. They are documented, filmed, sampled and studied. They exist in the deepest part of the Mariana Trench. Before we descend, take a small moment to notice where you are right now. Perhaps you are listening in the quiet of your bedroom, or through headphones on a late evening walk. Maybe it is early morning where you are, or long past midnight. Wherever you happen to be, you are many thousands of meters above a world that runs by entirely different rules. A world that does not know about clocks or schedules or streetlights. A world shaped by pressure, chemistry, and slow biological patience. If you find this journey calming, or if the idea of drifting slowly downward through layers of ocean feels like a good way to unwind, consider giving the video a like and perhaps subscribing. It helps this quiet corner of science reach others who might enjoy exploring the unseen parts of our planet at a gentler pace. Now, let us begin by imagining the descent. The Mariana Trench sits in the western Pacific Ocean, east of the Philippines. Its deepest point, known as Challenger Deep, reaches nearly 11,000 meters below sea level. To put that into perspective, if you were to take Mount Everest, the tallest mountain on Earth, and place it inside the trench, its peak would still be more than a mile underwater. That is how deep this place is. As you descend from the ocean surface, sunlight gradually fades. At around 200 meters, you enter what is called the twilight zone. Light is dim, colours drain away, and red becomes black. By 1,000 meters, you reach the midnight zone. No sunlight penetrates here. None. From this depth downward, the ocean is permanently dark. There is no sunrise, no sunset, just steady blackness. Continue downward, past 4,000 meters, into what scientists call the abyssal zone. Pressure increases constantly. At sea level, we live under 1 atmosphere of pressure. At 10,000 meters, pressure exceeds 1,000 atmospheres. That is roughly the equivalent of having a fully loaded freight train balanced on every square meter of your body. Metal bends, plastic cracks, airspaces collapse instantly. For a long time, scientists assumed complex life could not survive under such conditions. The reasoning seems straightforward. Cells contain structures that depend on precise chemical balances. Proteins fold into exact shapes. Membranes maintain stable boundaries. Extreme pressure distorts these structures. It interferes with molecular bonds. It squeezes water into places it does not normally occupy. On the surface, it seems incompatible with life as we know it. And yet, as deep-sea exploration advanced in the 20th century, especially with the development of reinforced submersibles and remote-operated vehicles, cameras began sending back images that did not match earlier assumptions. There were fish, there were crustaceans, there were organisms moving deliberately across the seafloor. One of the first surprises was simply that anything with a backbone could exist at such depth. Vertebrates have internal skeletons. They have tissues with air spaces. They have complex organ systems. All of that seems fragile under immense pressure. But certain species have adapted in ways that make them uniquely suited to the deep. The key lies partly in chemistry. deep-sea organisms often produce special molecules known as piezolites. These molecules stabilize proteins under high pressure, preventing them from unfolding or malfunctioning. In effect, they act as chemical support beams at the microscopic level. Without them, the proteins inside cells would distort and fail. Membranes also adapt. In shallow water species, cell membranes contain specific ratios of fatty molecules that maintain fluidity. At great depths, these ratios shift. Membranes remain flexible instead of becoming rigid under pressure. Flexibility is crucial. If membranes harden, cells cannot function properly. Many deep-sea fish lack swim bladders, the gas-filled organs that help shallow fish control buoyancy. Gas compresses under pressure, which would render such structures useless or dangerous at depth. Instead, these creatures rely on gelatinous tissues and carefully balanced body compositions. Some are almost neutrally buoyant, suspended in the water with minimal effort. There is also the matter of temperature. Deep ocean water is cold, typically between 1 and 4 degrees Celsius. Cold slows chemical reactions. It slows metabolism. Deep-sea organisms often grow slowly, reproduce infrequently, and live longer than their shallow counterparts. Energy is scarce, so nothing is wasted. Food does not fall from above in generous quantities. At the surface, sunlight drives photosynthesis, creating abundant plant life that supports complex food webs. In the trench, there is no sunlight. Life depends on what drifts down from above. Tiny particles of organic material, sometimes called marine snow. Occasionally, the body of a whale or large fish sinks to the bottom, creating a temporary feast that may last for years. But these events are rare. Some organisms rely on chemosynthesis instead of photosynthesis. In certain deep-sea environments, especially near hydrothermal vents, bacteria use chemical energy from compounds like hydrogen sulfide to produce organic matter. Larger organisms then feed on these bacteria or form symbiotic relationships with them. Entire ecosystems can form around chemical reactions rather than sunlight. It is in these extreme conditions that we encounter creatures that seem improbable. Consider the Mariana snailfish. At first glance, it does not look particularly dramatic. It is pale, almost translucent, with a soft body and small eyes. It does not have sharp teeth or glowing appendages. It looks delicate. That is precisely what makes it so surprising. This fish has been recorded at depths exceeding 8,000 meters. Its body lacks heavy bones. Instead, it is supported by cartilage. Its tissues are gelatinous, minimizing pressure differentials. It appears almost fragile, yet it is built specifically for a world that would crush sturdier forms. Then there are amphipods, small crustaceans related to shrimp. In the trench, some species grow larger than expected, a phenomenon sometimes referred to as deep-sea gigantism. These amphipods scavenge for food on the seafloor. They're tough, mobile, and surprisingly resilient. In one expedition, researchers lowered baited traps to extreme depths and recovered amphipods in significant numbers. Life was not sparse in the way early scientists imagined. It was active. Other fish in deeper zones possess oversized jaws and expandable stomachs. In an environment where meals are unpredictable, the ability to consume prey nearly as large as oneself is advantageous. A fish that can swallow a rare opportunity whole has better odds of surviving until the next. Bioluminescence also plays a role in the deep ocean. Many species produce light through chemical reactions involving molecules such as luciferin and enzymes like luciferase. Light can attract prey, deter predators, or facilitate communication. In total darkness, even a faint glow stands out. Some fish have photophores arranged in patterns along their bodies. Others dangle glowing lures in front of their mouths. And still, much of the trench remains unexplored. Only a handful of crude descents have reached its deepest point. Robotic vehicles have expanded our view, but the ocean is vast. Every expedition tends to reveal species never before documented. New worms, new crustaceans, new microbial communities. What makes these discoveries striking is not merely their novelty, but the reminder that life adapts in ways that challenge assumptions. Conditions that appear hostile from our perspective may be entirely normal from another organism's viewpoint. Pressure, darkness, cold, chemical gradients. These are not obstacles in the same way for a trench-dwelling fish as they are for a human. And so, as we continue downward in our exploration, moving past the broad overview and into the specific forms that inhabit this environment, it becomes clear that the phrase "shouldn't exist" says more about our expectations than about the organisms themselves. Biology does not follow human intuition. It follows chemistry, physics, and the gradual shaping force of evolution across immense spans of time. Down here, in the deepest folds of Earth's crust, evolution has had millions of years to experiment quietly. No sunlight, no storms, no seasonal change, just pressure, darkness, and slow persistence. And in that stillness, bodies have taken on forms that look improbable when viewed through the lens of surface life. As we move closer to the seafloor itself, where sediment settles like fine dust and the water above weighs down with relentless force, we begin to encounter the first of these truly unusual inhabitants in more detail, creatures whose very structure forces us to reconsider what is biologically possible under conditions that, by all surface standards, seem entirely unlivable. If you picture the Earth from a distance, oceans appear smooth and continuous, vast blue surfaces stretching across most of the planet. From space, they look calm, almost uniform. It is easy to imagine them as deep but featureless reservoirs of water. The reality beneath that surface is far more complex. The ocean floor has mountains, ridges, plains, volcanoes, and trenches deeper than any valley on land. And among all those formations, the Mariana Trench stands as the deepest known natural point on Earth. It sits in the western Pacific Ocean, east of the Mariana Islands, formed by the slow but relentless movement of tectonic plates. The Pacific Plate is sliding beneath the smaller Mariana Plate in a process called subduction. Over millions of years, this downward motion has carved an enormous crescent-shaped trench in the seafloor. The deeper section, Challenger Deep, reaches close to 11,000 meters below sea level. Numbers like that are hard to visualize, so comparisons help. Commercial airplanes cruise at roughly 10,000 meters above the Earth. If one could somehow fly directly downward instead of forward, it would barely reach the trench floor before running out of altitude. Despite that extreme depth, the trench is not a simple vertical drop. It has slopes, terraces, ridges, and sediment fields. Some areas descend gradually, others plunge sharply. The terrain changes from rocky outcrops to soft sediment plains where fine particles settle over thousands of years. These particles come from many sources: microscopic organisms that live near the surface, volcanic ash, eroded continental material carried by currents. Everything eventually sinks. Because sunlight cannot reach those depths, visualizing the trench requires instruments rather than direct observation. Sonar mapping has played a major role. Ships emit sound pulses downward. The time it takes for the echo to return reveals depth. Over decades, increasingly precise sonar surveys have refined our understanding of trench topography. Early measurements varied widely. Some estimates differed by hundreds of meters. Today, mapping is far more accurate, though not perfect. The ocean still holds mapping uncertainties comparable to unexplored land regions centuries ago. Direct visits to Challenger Deep are rare. The first occurred in 1960 when Jacques Picard and Don Walsh descended in the bathyscaphe Trieste. The vessel was essentially a reinforced sphere suspended beneath a buoyant float filled with gasoline, which resists compression better than air. Their descent took nearly 5 hours. At the bottom, visibility was limited because the craft stirred up sediment. They stayed about 20 minutes before beginning the long ascent. Even that brief visit proved something significant. They reported seeing a flatfish-like creature, though later interpretations suggest it may have been a sea cucumber or another invertebrate. Still, the observation challenged the assumption that complex life could not survive there. Decades passed before another crude descent. Technological limitations, cost and risk all played roles. Pressure at those depths reaches more than 1,000 times surface atmospheric pressure. Every seal, every viewport, every structural element must tolerate that stress. A small flaw could lead to catastrophic failure. Material science has gradually improved, allowing newer submersibles to revisit the trench. Filmmaker James Cameron made a solo descent in 2012 using the Deep Sea Challenger submersible, collecting samples and high-definition footage. Since then, a handful of additional missions have taken place, though they remain exceptional events. Most exploration now relies on remotely operated vehicles and autonomous landers. These machines descend tethered to research vessels or operate independently, recording video, collecting sediment cores, and capturing biological specimens. Cameras designed for extreme pressure have revealed landscapes that look calm but alien. Fine sediment ripples slightly with currents. Small crustaceans move across the bottom. Occasionally fish glide past, slow and deliberate. One striking aspect of the trench environment is its stability. On land, weather constantly reshapes surfaces. Rain erodes rock. Wind redistributes sand. Temperature cycles crack materials. In the deep trench, conditions change slowly. Temperature remains consistently cold. Darkness is constant. Currents exist but are often gentle. Geological change occurs, but on timescales that stretch far beyond human observation. That stability influences biology. Species living there often evolve specialized traits without the pressure of rapid environmental shifts. At the same time, isolation limits genetic exchange. Populations may remain separated for long periods, allowing distinct evolutionary paths. Some trench organisms appear closely related to species found in shallower depths, but show specific adaptations: altered body chemistry, reduced skeletal density, modified sensory systems. Sound behaves differently down there too. Water transmits sound efficiently, and in the absence of surface noise, certain frequencies travel long distances. Some deep-sea organisms are sensitive to vibration rather than visual cues. Vision has limited usefulness where light never arrives. Eyes may be small, reduced, or adapted to detect faint bioluminescence. Other senses compensate. Chemistry also plays a defining role. Sediments contain organic material that slowly decomposes. Microbial communities thrive on these processes, recycling nutrients. In certain areas, chemical gradients create localized ecosystems. Hydrothermal vents, though not dominant in Challenger Deep itself, exist elsewhere along subduction zones. They release mineral-rich fluids that support chemosynthetic bacteria. Those bacteria in turn sustain larger organisms. It is worth noting that the trench is not entirely cut off from the rest of the ocean. Water circulates, though slowly. Organic material sinks from above. Dead plankton, fish waste, fragments of algae. Over long periods, this constant drizzle accumulates. Scientists sometimes refer to it informally as marine snow. Without it, many trench ecosystems would lack sufficient energy input. Human influence has reached even these depths. Plastic debris has been documented in deep-sea samples. Chemical pollutants have been detected in amphipods collected from the trench floor. Persistent organic pollutants, once released into the environment, circulate globally. Ocean currents distribute them widely. Their presence in one of the most remote places on Earth is a reminder of how interconnected planetary systems are. Temperature remains near freezing, typically just above 0 Celsius. Cold slows metabolism. Organisms conserve energy. Movement tends to be deliberate. Rapid bursts of activity are rare. Food scarcity reinforces this pattern. Efficient energy use becomes essential for survival. Another factor shaping trench life is pressure-induced solubility changes. Gases dissolve more readily under high pressure. Oxygen availability can differ from expectations based on shallow water measurements. Some organisms possess specialized respiratory proteins that function effectively under these conditions. Hemocyanin and hemoglobin variants exhibit pressure tolerance. These biochemical adaptations are subtle but critical. Research into deep-sea pressure tolerance has practical applications. Enzymes from piezophilic organisms, those adapted to high pressure, interest industrial and medical researchers. Understanding how proteins remain stable under compression could inform biotechnology. Deep-sea microbes also produce unique compounds, some with antimicrobial properties. Exploration of extreme environments sometimes yields unexpected benefits far removed from oceanography. Navigation in the trench environment presents challenges. GPS signals do not penetrate water effectively. Submersibles rely on inertial navigation systems, acoustic positioning, and careful tether management. Communication with surface vessels is slow. Acoustic signals travel at roughly 1,500 meters per second in seawater, so even simple commands involve noticeable delay at extreme depth. Lighting for observation requires powerful lamps. Without them, cameras would record only darkness. Artificial light can alter animal behavior, so researchers try to minimize disturbance. Red light, less visible to many deep-sea species, is sometimes used. Still, any illumination introduces an element foreign to that environment. Sediment composition varies. Some areas contain fine clay-like particles, others include coarser material, shell fragments, mineral grains. Sediment cores extracted by landers reveal layered histories. Each layer corresponds to different deposition periods, volcanic events, climatic shifts. Studying those layers helps reconstruct Earth's environmental past. Seismic activity occasionally affects the trench. Subduction zones are associated with earthquakes. When seismic events occur, sediment may shift. Landslides can transport material downslope. Such disturbances alter local habitats. Organisms adapted to stability must cope with episodic disruption. Water chemistry includes elevated carbon dioxide concentrations compared to surface waters. Combined with cold temperatures, this influences acidity. Some organisms possess shells or exoskeletons that resist dissolution despite these conditions. Others avoid calcified structures altogether, relying on softer tissues. Despite technological progress, large portions of the trench remain unobserved directly. Mapping resolution still leaves gaps. Biological surveys sample limited areas. Each expedition covers only a fraction of the total trench length. Estimates of species diversity remain tentative. Many organisms likely remain undocumented. Curiosity about the trench extends beyond marine biology. Planetary scientists study extreme Earth environments as analogues for extraterrestrial conditions. High pressure, absence of sunlight, chemical energy-based ecosystems. These factors resemble hypothetical environments on icy moons like Europa or Enceladus. If life exists there, it may share functional similarities with trench organisms. The psychological aspect of descending into such depths also draws attention. Human explorers report an unusual sense of isolation. Outside the viewport, nothing but darkness and occasional particulate drift. Communication delays reinforce separation from the surface. It is not dangerous in the immediate sense when equipment functions correctly, but it is profoundly different from any familiar environment. Equipment reliability remains critical. Pressure-resistant housings, redundant systems, careful engineering margins. Even then, missions carry inherent risk. Each descent involves extensive planning, testing, and contingency protocols. That level of preparation partly explains why visits are infrequent. Still, interest continues to grow. Advances in robotics, material science, and imaging technology gradually lower barriers. New expeditions aim not only to document life, but to understand ecological relationships, chemical cycles, geological processes. The trench serves as a natural laboratory for extreme condition research. As observations accumulate, earlier assumptions continue to shift. Life is not absent. It is not even necessarily sparse in every location. Instead, it is specialized. Adaptations that seem unusual from a surface perspective become logical within trench conditions. Soft bodies reduce pressure differentials. Biochemical stabilizers protect cellular structures. Energy efficiency replaces speed. All of this forms the backdrop for understanding the creatures themselves. Before focusing on individual species, recognizing the environment they inhabit clarifies why their features appear unconventional. Nothing there developed randomly. Each trait reflects millions of years of gradual adjustment to pressure, darkness, cold, and limited food supply. So when unusual fish glide slowly past a camera lens, or translucent crustaceans cluster around baited traps, they are not anomalies in their own world. They are residents perfectly suited to it. From our perspective, they may look improbable. From theirs, conditions are ordinary. And as exploration pushes further into these depths, revealing more organisms with unfamiliar forms, the trench continues to function as a reminder that Earth still contains frontiers not fully charted. Not distant planets, not theoretical environments, but a region on our own world, quietly existing beneath kilometers of water, shaping life in ways that remain partly hidden, gradually becoming clearer with each careful descent, each new sample, each additional glimpse into that deep, pressurized landscape where evolution has been working steadily, largely unnoticed for a very long time. As the descent continues deeper into the Mariana Trench, one factor becomes impossible to ignore. Pressure increases steadily, quietly, without drama, yet with overwhelming force. At the ocean's surface, the air pressing down on you equals roughly 1 atmosphere. It is something you rarely think about. It is simply there. But underwater, pressure rises fast. Every 10 meters of depth adds another atmosphere. By the time you reach the deepest parts of the trench, pressure exceeds 1,000 atmospheres. That is not a metaphorical description. It is a physical load strong enough to crush steel structures not specifically engineered to withstand it. If a typical human body were suddenly transported there without protection, collapse would be immediate. Air spaces in the lungs, sinuses, and tissues would compress violently. Cellular structures would fail. Proteins would deform. Nerve signals would cease. The idea that animals not only survive but actively live under such conditions initially seemed unlikely to early oceanographers. The assumption was reasonable. Life on land and in shallow water depends heavily on stable pressure ranges. Most biological systems evolved within narrow limits. Yet the trench demonstrates that biology can operate under conditions far outside everyday human experience. The key lies in gradual adaptation across immense spans of evolutionary time. Nothing in the trench appeared suddenly. Each organism represents countless generations adjusting incrementally, selecting for traits that improve survival under crushing pressure. To understand why pressure matters so much, it helps to think at the molecular level. Cells rely on proteins for nearly every function. Proteins fold into precise three-dimensional shapes. Those shapes determine how enzymes catalyze reactions, how receptors bind molecules, how structural elements maintain integrity. Pressure interferes with folding. It forces water molecules into spaces within protein structures, destabilizing them. Under extreme compression, many proteins simply stop working. Deep-sea organisms counter this through specialized biochemical compounds called piezolites. These molecules stabilize proteins, preventing deformation. One commonly studied example is trimethylamine N oxide, often abbreviated as TMAO. It accumulates in the tissues of deep-sea fish. Its concentration tends to increase with depth. Essentially, it acts as a molecular brace, helping proteins maintain functional shapes despite external compression. Interestingly, this adaptation has limits. Researchers believe that beyond certain depths, the amount of TMAO required would become biologically impractical. That may partly explain why vertebrate fish appear absent below certain extreme thresholds. while invertebrates and microbes continue deeper. Chemistry imposes boundaries even on highly adaptable systems. Cell membranes present another challenge. These membranes consist largely of lipid molecules arranged in bilayers. Their fluidity is essential. If membranes become too rigid, transport processes fail. Nutrients cannot enter efficiently. Waste products accumulate. Pressure tends to compress membranes, reducing fluidity. Deep-sea organisms adjust lipid composition to compensate. They incorporate fatty acids that maintain flexibility under compression. It is a subtle shift, invisible without biochemical analysis, yet crucial. Gas-filled structures create additional complications. Many shallow water fish possess swim bladders that regulate buoyancy. Gas compresses dramatically under pressure, making such organs unreliable or dangerous at depth. Most trench-dwelling fish lack them entirely. Instead, they rely on lipid storage, reduced skeletal mass, and gelatinous tissues to achieve neutral buoyancy. Their bodies often appear soft, even fragile, but that softness reduces differential compression. Bone density itself can be problematic. Heavy mineralized skeletons resist compression poorly compared to flexible cartilage-like structures. Several deep-sea fish species display reduced ossification. Their skeletons are lighter, more compliant. Again, this looks delicate from a surface perspective, but proves advantageous in a high-pressure environment. Metabolism slows significantly in the trench. Pressure influences enzyme kinetics. Combined with cold temperatures, reaction rates decrease. Organisms adapt by conserving energy. Growth tends to be slow. Reproduction may occur less frequently. Longevity sometimes increases. These patterns reflect energy scarcity as much as pressure effects, but the two factors interact closely. At first glance, you might expect pressure alone to determine survivability. The reality is more complex. Temperature, oxygen availability, nutrient supply, and chemical composition all contribute. Pressure acts as a constant background condition shaping how these other factors manifest. Organisms must integrate multiple adaptations simultaneously. Consider microbial life. Bacteria known as piezophils or barophiles thrive under high pressure. Some actually require it. When brought to surface conditions in laboratories, they function poorly or die. Their enzymes evolve specifically for compressed environments. Studying these microbes has provided insights into protein stability, membrane chemistry, and potential industrial applications. Certain deep-sea bacteria participate in nitrogen cycling, sulfur metabolism, and methane oxidation. These processes influence global biogeochemical cycles. Even though the trench seems remote, microbial activity there contributes to planetary chemistry. Carbon sequestration, nutrient recycling, and chemical fluxes link deep environments to surface ecosystems indirectly. Invertebrates demonstrate equally fascinating adaptations. Amphipods, for example, possess exoskeletons composed partly of chitin reinforced with minerals. Some trench amphipods incorporate aluminum compounds from sediments into their exoskeletons. Researchers believe this may provide additional structural resilience under pressure. The exact mechanisms remain under investigation, but the observation highlights how environmental chemistry influences biological design. Deep-sea worms, sea cucumbers, and other benthic organisms often have hydrostatic skeletons rather than rigid frames. Their bodies rely on fluid pressure within tissues for support. External pressure equalizes internal pressure, reducing structural strain. Flexibility becomes an asset rather than a weakness. Sensory systems adapt too. Vision loses importance in permanent darkness, but mechanoreception becomes critical. Detecting vibrations helps locate prey, avoid predators, and navigate terrain. Pressure transmits vibrations efficiently through water, so mechanosensory organs function effectively. Some fish possess lateral line systems highly tuned for this purpose. Bioluminescence appears again as a useful adaptation. Producing light under extreme pressure requires enzymes and substrates stable under those conditions. Luciferous systems in deep-sea organisms exhibit pressure tolerance. Light serves multiple roles: communication, camouflage, prey attraction. Counterillumination, where organisms emit light matching faint downwelling light, helps conceal silhouettes from predators below in slightly shallower zones. Even near trench depths, bioluminescence remains widespread. Reproductive strategies adjust as well. Low population densities mean encounters between potential mates are infrequent. Some species rely on chemical signaling over distance. Others exhibit hermaphroditism, increasing reproductive flexibility. In certain anglerfish species, though more common in upper deep zones than the trench floor, males attach permanently to females, ensuring reproductive access. While not universal in the trench itself, the principle illustrates how environmental constraints shape reproduction. Embryonic development under pressure presents challenges. Cell division, differentiation, and growth processes must tolerate compression. Egg cases, protective membranes, and biochemical stabilizers all play roles. Some species produce fewer but more resilient offspring rather than large numbers of vulnerable ones. Laboratory simulation of trench pressure remains difficult. High-pressure chambers exist, but maintaining stable biological conditions simultaneously is technically demanding. Sample retrieval often alters organisms before study. Rapid decompression can damage tissues. Specialized pressure-retaining containers help mitigate this, but are not always feasible. Because of these challenges, much knowledge comes from indirect observation, video footage, genetic sequencing from environmental DNA, chemical analyses of sediments. Each method contributes partial information. Integrating them gradually clarifies the picture. Pressure also influences sound propagation. Acoustic communication, if present, would behave differently compared to shallow waters. Research continues into whether certain deep organisms use pressure-stable acoustic signals intentionally or simply respond to ambient vibrations. There is also the question of limits. How deep can complex life persist? Challenger. Deep marks the deepest known oceanic environment, but theoretical considerations extend further. Subsurface ocean worlds on icy moons may experience comparable or greater pressures beneath ice crusts. Studying trench organisms provides analogues for astrobiology. If life adapts here, similar biochemical strategies might function elsewhere. Engineering lessons emerge as well. Materials designed to withstand high pressure, pressure-resistant electronics, flexible structural components. Biomimicry occasionally draws inspiration from deep-sea adaptations. Nature often solves problems engineers later encounter. Human exploration remains cautious. Submersibles undergo rigorous testing. Hull shapes favour spheres because pressure distributes evenly across curved surfaces. Viewports use thick acrylic or specialised glass. Redundancy in life support systems is essential. Even with precautions, missions involve risk. Psychologically, pressure is invisible. Descending occupants feel no crushing sensation inside properly pressurised cabins. The knowledge of external force creates a different kind of awareness. Outside, thousands of tons of water press inward. Inside, relative calm persists. That contrast underscores the importance of engineering precision. As research expands, it becomes increasingly clear that life's adaptability extends far beyond earlier expectations. The phrase "should collapse" reflects assumptions based on human physiology. Organisms evolving entirely under high pressure experience it as normal baseline rather than stressor. Their biochemical frameworks incorporate compression as a constant. So when cameras capture translucent fish hovering just above sediment, or amphipods scuttling calmly across the trench floor, what appears improbable from a surface viewpoint represents successful adaptation in context. These creatures do not resist pressure in the way a human diver resists depth. They embody it. Their chemistry, structure, metabolism, and behaviour integrate seamlessly with their environment. And as the exploration continues deeper into specific species, examining their anatomy, feeding strategies and ecological relationships, the role of pressure remains present in every detail, shaping bodies, influencing survival strategies, quietly defining the rules of existence in one of Earth's most extreme and least familiar habitats. As the descent continues past the depths where even faint traces of sunlight disappear, another defining condition takes over completely: darkness. Not the kind experienced at night on land where distant city glow, moonlight or starlight soften the edges. This is absolute absence of sunlight, a permanent state that has existed for millions of years. In the deepest parts of the Mariana Trench, light from the sun simply never arrives. It is absorbed by the upper layers of the ocean long before reaching those depths. Sunlight fades gradually as you go down. The upper few hundred meters still carry usable light. Photosynthesis occurs. Colours remain visible. But after about 1,000 meters, the ocean enters what scientists often call the midnight zone. Below that, darkness becomes constant. The trench floor sits far deeper than that threshold. No dawn, no dusk, no seasonal variation in light. Just steady blackness interrupted only by occasional biological light or artificial illumination from research equipment. For life that evolved under these conditions, darkness is not an obstacle in the same way it would be for humans. It is simply the normal environment. Still, the absence of sunlight has far-reaching consequences. The most obvious is the complete lack of photosynthesis. On land and in shallow seas, sunlight drives plant growth, which supports herbivores, which in turn support predators. Remove sunlight and that primary energy pathway disappears. The trench ecosystem must rely on different energy sources. One major source is organic material sinking from above. Microscopic plankton, fragments of algae, waste products, and the occasional larger carcass descends slowly through the water column. By the time this material reaches trench depths, much of it has decomposed or been consumed. What remains forms a thin, continuous drizzle, sometimes called marine snow. It is not snow in the frozen sense, but the visual analogy fits. Tiny particles drifting downward steadily. This limited food supply shapes biological strategies. Organisms conserve energy. Movement is often slow and deliberate. Many species exhibit opportunistic feeding habits. If something edible appears, it is rarely ignored. Some fish have expandable stomachs, allowing them to consume prey larger than expected relative to their body size. Such flexibility improves survival when meals are unpredictable. Darkness also reshapes sensory priorities. Vision becomes less useful unless organisms generate their own light. Many trench creatures have reduced eyes, or eyes adapted specifically to detect bioluminescence. Others rely more heavily on non-visual senses. Mechanoreception, the ability to detect vibrations in water, becomes crucial. Chemical sensing also plays a major role. Dissolved molecules can signal food sources, predators, or potential mates. Bioluminescence emerges as one of the most distinctive features of deep-sea life. Chemical reactions within specialized organs produce light without heat. The most common mechanism involves luciferin reacting with oxygen in the presence of luciferase enzymes. Variations exist, but the principle remains similar. Light emission can be continuous, pulsed, or triggered by external stimuli. Functions of bioluminescence vary. Some organisms use light to attract prey. Anglerfish, though typically found slightly shallower than the deepest trench floor, are well-known examples. A glowing lure extends from the head, drawing curious animals closer. Others use light defensively. Sudden flashes can startle predators or create confusion. Counter-illumination provides camouflage in certain depth ranges. Organisms emit light downward, matching faint residual light from above, reducing their silhouette when viewed from below. While Challenger Deep receives essentially no sunlight, Species inhabiting slightly higher zones benefit from this adaptation. Communication represents another possible function. Patterns of flashes may signal reproductive readiness or territorial presence. Research continues, as observing natural behavior without disturbance remains difficult. Artificial lighting from submersibles can alter responses. Coloration becomes less relevant without light. Many deep-sea animals appear pale, translucent, or darkly pigmented. Red pigmentation, which appears black in low light, is common in some species because red wavelengths disappear quickly with depth. A red animal becomes effectively invisible. Structural adaptations often accompany sensory changes. Eyes may enlarge to capture faint light or reduce entirely to conserve energy. Olfactory organs can become more sensitive. Lateral line systems in fish detect water movement with high precision. These systems allow navigation and hunting without visual cues. Circadian rhythms, the biological cycles tied to day-night patterns on the surface, function differently in perpetual darkness. Some deep-sea species maintain internal rhythms independent of sunlight. Others appear less strongly tied to daily cycles. Research suggests feeding availability, tidal influences, and chemical gradients may act as alternative timing cues. Temperature stability contributes to environmental consistency. Without sunlight-driven heating cycles, temperatures remain near freezing with minimal fluctuation. This stability simplifies some physiological regulation but reinforces slow metabolic rates. One question often arises about plant life in such darkness. Traditional plants relying on photosynthesis cannot survive there. However, microbial communities using chemosynthesis thrive in certain deep environments, particularly near hydrothermal vents or cold seeps. They convert chemical energy into organic matter. Larger organisms sometimes host these microbes symbiotically, effectively outsourcing primary production. Sediment composition reflects long-term deposition in darkness. Without photosynthetic plants stabilizing sediments, particle accumulation depends on external inputs. Layers build slowly. Disturbances from currents or seismic activity occasionally redistribute material. Predation strategies adapt to low visibility. Ambush tactics become common. Some predators remain motionless until prey approaches. Others use chemical cues to track food sources. Fast pursuit is less common due to energy costs and limited visibility. Reproductive encounters pose challenges. Sparse populations and darkness reduce chance encounters. Chemical signaling over distance helps. Some species release pheromones detectable through sensitive receptors. Others synchronize reproductive timing based on environmental cues unrelated to light. Egg development occurs in darkness as well. Protective membranes and biochemical stabilizers ensure embryos develop successfully without sunlight. Some species brood eggs internally, reducing exposure risks. Human exploration highlights the psychological impact of total darkness. Submersible occupants report unusual perceptions when external lights switch off. Without visual reference points, orientation becomes difficult. The sense of isolation intensifies. These experiences underscore how strongly human perception depends on light. Technological lighting used in exploration introduces temporary artificial brightness. Cameras require illumination, but researchers attempt to minimize ecological disruption. Red light, less visible to many deep-sea species, sometimes serves observational purposes. Even so, artificial light remains foreign to the natural environment. Acoustic mapping substitutes for visual surveying. Sonar pulses reveal terrain shapes invisible to cameras without lighting. This technology has mapped large portions of the trench floor. The resolution varies. Darkness influences decomposition rates. Without ultraviolet radiation and with low temperatures, certain materials degrade slowly. Organic remains may persist longer than at the surface. This provides intermittent food sources for scavengers. Human-generated debris has unfortunately reached even these depths. Plastic fragments and synthetic fibers have been documented in trench sediments. Darkness does not prevent pollutant accumulation. Ocean currents distribute contaminants widely. Microbial activity continues regardless of light absence. Bacteria break down organic matter, recycle nutrients, and contribute to chemical cycles. These processes sustain broader ecosystems indirectly. Evolutionary pressures in darkness emphasize efficiency. Extraneous features tend to diminish over generations. Energy conservation dominates. Adaptations that might seem extreme from a surface viewpoint simply reflect optimization under constant low-light conditions. There is also a philosophical dimension often mentioned by researchers. A world without sunlight challenges intuitive associations between light and life. Surface ecosystems depend heavily on solar energy. The trench demonstrates that life can persist using alternative energy pathways, albeit with different structures and slower dynamics. Comparisons with extraterrestrial environments arise again. Subsurface oceans beneath ice-covered moons lack sunlight entirely. Studying trench organisms provides insight into how life might function under similar conditions elsewhere. Navigation for trench animals relies on gradients rather than landmarks. Chemical gradients, pressure gradients, current patterns. These cues guide movement in the absence of visual scenery. Some species exhibit site fidelity, remaining within specific zones for extended periods. Growth patterns reflect limited energy input. Many organisms grow slowly but steadily. Longevity may increase accordingly. Exact lifespans remain difficult to measure, but some deep-sea species show evidence of extended life cycles. Despite perpetual darkness, biological light ensures the trench is not entirely devoid of illumination. Brief flashes, steady glows, intermittent signals. From a human perspective, it would appear as scattered points of faint light in otherwise complete blackness. Research continues to uncover new species exhibiting unique light-producing mechanisms. Genetic analyses reveal diversity in luciferin-luciferous systems. Some organisms acquire luminescent bacteria symbiotically rather than producing light independently. All these factors combine to create an ecosystem fundamentally different from sunlit environments, yet fully functional on its own terms. Darkness defines energy flow, sensory systems, behavior, and evolutionary pathways. It shapes the creatures encountered there just as strongly as pressure does. And as attention shifts toward the organisms themselves, examining their bodies, behaviors, and survival strategies, the constant absence of sunlight remains an underlying influence, quietly structuring every adaptation observed in that deep, unlit expanse where life continues steadily, despite conditions that, from the surface perspective, might initially seem incompatible with complex biological existence. As exploration vehicles drift slowly above the trench floor, their lights sometimes catch the outline of something that does not immediately look solid. At first, it can resemble a ripple in the water, a distortion rather than a body. Then the shape becomes clearer. Fins move gently. A head turns. There is a fish there, but it appears almost transparent, its tissues pale and gelatinous, its structure faintly visible through its own skin. In a place defined by darkness and pressure, some of the most remarkable residents are fish that seem barely there at all. One of the most well-known examples associated with the deepest parts of the Mariana Trench is the Mariana snailfish, a member of the family Lipariae. These fish have been recorded at depths exceeding 8,000 meters, making them among the deepest living vertebrates known. They do not look powerful or armored. They look soft. Their bodies are lightly built, their skeletons reduced, their muscles modest. When filmed by deep-sea cameras, they move slowly, almost gently, hovering just above the sediment. The transparency of some deep-sea fish serves multiple purposes. In a world without sunlight, camouflage still matters. Bioluminescence from other organisms can create brief flashes of light. Any solid silhouette becomes visible in that moment. Transparent or semi-translucent tissues reduce contrast. Instead of appearing as a defined shape, the fish blends into the surrounding water. Pigmentation costs energy to produce. In an environment where food is limited, reducing unnecessary biological processes becomes advantageous. Many trenchfish lack the vibrant colours seen in coral reefs or shallow seas. Their skin appears pale, sometimes milky, sometimes faintly pink due to underlying blood vessels. In some cases, internal organs can be partially seen through the body wall. The snailfish's body composition reflects adaptation to both pressure and darkness. Their tissues contain high water content, creating a gelatinous consistency. This reduces density differences between body and surrounding water, aiding buoyancy without the need for a gas-filled swim bladder. As mentioned earlier, swim bladders are impractical at extreme depths because gas compresses under pressure. By relying on tissue composition rather than gas regulation, these fish maintain neutral buoyancy more reliably. Their skeletons are lightly ossified. Bones are thin, sometimes partially cartilaginous. A heavy rigid skeleton would add unnecessary mass and create stress points under compression. Flexibility distributes pressure more evenly. The result is a fish that looks fragile but is chemically and structurally suited to its environment. Eyes in these fish vary in size. Some deep snailfish possess relatively small eyes compared to shallow relatives. Vision remains useful for detecting bioluminescent flashes, but the absence of ambient light reduces the need for complex visual systems. Energy saved from reduced eye development can support other functions. Their mouths are modest compared to the exaggerated jaws of certain other deep-sea predators. Snailfish often feed on small crustaceans, amphipods, and invertebrates found on or near the seafloor. Video footage from baited landers has shown them approaching traps calmly, feeding without apparent urgency. Their movements suggest energy conservation rather than aggressive hunting. Biochemical adaptation remains central to their survival. High concentrations of TMAO stabilize their proteins under pressure. Research indicates that TMAO levels increase predictably with depth in many fish species. This compound offsets pressure-induced destabilization of enzymes and structural proteins. However, TMAO also affects osmotic balance. Maintaining proper internal chemistry requires careful regulation. Reproduction among deep snailfish remains partly mysterious due to limited observation. Some related species lay egg masses attached to substrates, others may brood eggs internally. In the trench, sparse populations mean locating mates can be challenging. Chemical signalling likely plays a role. Detailed life cycle studies remain ongoing. Another example of translucent deep-sea fish includes certain species of cusk eels and eelpouts found in abyssal zones. While not always as deep as the Mariana snailfish, they exhibit similar gelatinous tissues and reduced pigmentation. Their elongated bodies allow them to navigate close to the sediment, searching for small prey. Transparency also influences predation risk. Larger predators in the trench are fewer compared to shallower depths, but they exist. A fish that presents minimal visual signature during brief bioluminescent illumination reduces its chances of detection. This is especially relevant in mid-water regions above the trench floor, where occasional light from other organisms may reveal silhouettes. The internal anatomy of these fish often reflects simplification. Swim bladders are absent. Some organ systems are reduced in complexity compared to shallow water relatives. Energy efficiency governs much of their design. Muscles sufficient for slow hovering and short movements replace those needed for rapid bursts of speed. Scientists studying captured specimens must account for decompression effects. Bringing a fish from extreme depth to the surface can cause tissue damage if pressure is not maintained. Specialized pressure-retaining containers help preserve specimens close to their native state for laboratory analysis. Even then, changes can occur. Genetic analysis reveals that many trenchfish evolved from shallower ancestors. Over evolutionary time, certain populations adapted progressively to deeper habitats. Mutations that improved protein stability, membrane flexibility, and metabolic efficiency under pressure accumulated. Eventually, distinct species emerged specialized for life at extreme depth. Interestingly, there appears to be a depth limit for vertebrate fish survival. Beyond approximately 8,200 to 8,500 metres, vertebrates become extremely rare or absent. Invertebrates and microbes continue deeper. The biochemical demands of maintaining vertebrate physiology under higher pressures may approach impractical limits, particularly regarding TMAO accumulation. This potential boundary highlights the interplay between chemistry and evolution. The physical appearance of these ghost-like fish can appear almost unfinished to human eyes. Without bright coloration or rigid form, they resemble soft outlines more than solid creatures. Yet this design reflects precision adaptation rather than deficiency. Every aspect serves a purpose within trench conditions. Behavioural observations indicate that snailfish and similar species exhibit territorial tendencies near bait sources. Multiple individuals may gather around food, but maintain spacing. Their interactions appear subdued rather than aggressive. Energy conservation likely discourages unnecessary conflict. Their diet primarily consists of small crustaceans abundant in trench sediments. Amphipods often swarm baited cameras, and snailfish follow. This relationship suggests a predator-prey dynamic shaped by marine snow input and occasional larger carcasses. Because trench ecosystems receive limited energy, population densities remain relatively low compared to surface reefs. Transparent bodies may also reduce the need for heavy armour. With fewer visually hunting predators, selection pressure for thick protective scales diminishes. Instead, chemical and structural resilience against pressure takes priority. Studying these fish expands understanding of vertebrate adaptability. For decades, scientists doubted vertebrates could survive at such depths. Discoveries of Mariana snailfish challenged that assumption. Continued exploration may refine knowledge further, but current evidence suggests vertebrates approach physiological limits near these depths. There is also educational value in observing how perception shapes expectation. To a surface observer, a soft, translucent fish appears vulnerable. In the trench, vulnerability depends on different metrics: resistance to pressure, biochemical stability, efficient energy use. These traits define success. The slow, almost drifting movements of these fish contribute to their ghost-like impression. Without rapid swimming or dramatic behaviour, they seem to float rather than propel themselves. Their fins move in small adjustments. The water around them remains largely undisturbed. Artificial lights from research vehicles illuminate them briefly, before fading back into darkness as the vehicle moves on. In their natural environment, they would rarely experience such brightness. Their world consists of dim flashes from other organisms and the constant weight of water above. Every observation of these fish reinforces a broader pattern. Extreme environments do not eliminate complexity. They reshape it. Bodies become softer, colours fade, eyes adjust. Biochemistry compensates. Evolution continues working within constraints, producing forms that challenge surface-based intuition. As attention shifts further toward other unusual trench inhabitants, including larger crustaceans and bioluminescent organisms, the ghost-like fish remain an important reminder that vertebrate life extends deeper than once believed, adapting quietly and effectively in a realm defined by darkness, cold, and crushing pressure, where transparency becomes not a sign of weakness, but a practical solution to the unique demands of existence so far beneath the sunlit surface. As research lights fade into the distance and the deep water settles again into its natural state, the darkness of the trench becomes complete once more. There is no sunlight, no moonlight, no faint horizon glow. And yet, the darkness is not entirely empty. Every so often, a tiny flicker appears, sometimes a steady glow, sometimes a brief flash that vanishes almost as quickly as it came. These signals do not come from outside the ocean. They are produced by the creatures living there. In a world without sunlight, many organisms have developed the ability to create their own light. This ability, known as bioluminescence, is widespread throughout the deep ocean. It appears in fish, crustaceans, jelly-like organisms, worms, bacteria, and many other life forms. In shallower waters it can already be impressive, but in the deepest regions near the Mariana Trench, where darkness is permanent, biological light takes on particular importance. It becomes a tool for survival rather than a novelty. The basic chemistry behind bioluminescence is relatively consistent across species, even though the exact molecules can vary. A light-producing molecule, commonly referred to as luciferin, reacts with oxygen in a process catalyzed by an enzyme called luciferase. The reaction releases energy in the form of visible light. Unlike incandescent light sources, this process generates minimal heat. That efficiency is essential in cold environments where wasting energy could reduce survival chances. Different organisms emit different wavelengths of light. Blue and green light dominate because those wavelengths travel farthest through seawater. Red light fades quickly with depth. Some organisms produce bluish flashes, others green glows. Rarely species emit red light, which can function as a kind of private illumination, because many deep-sea animals cannot detect red wavelengths effectively. The reasons for producing light vary. One of the most common uses is attracting prey. Certain fish possess luminous organs positioned near their mouths. Small animals drawn to the glow approach what appears to be a potential food source or environmental signal. Instead, they encounter a predator. Anglerfish provide a familiar example, although many anglerfish live somewhat shallower than the trench floor. Still, similar strategies exist at greater depths. Another use is defense. Sudden flashes can startle predators long enough for escape. Some organisms release luminous clouds into the water when threatened. The glowing particles create visual confusion, sometimes compared to a marine equivalent of smoke screens. Predators focus on the distraction while the prey moves away slowly and quietly. Communication represents another possibility. While still under study, evidence suggests certain species use patterned flashes during reproductive periods. In environments where visual landmarks are absent, light signals may help individuals locate each other. Chemical signals likely play a role as well, but bioluminescence offers a more immediate directional cue. Camouflage through counter-illumination also occurs. In slightly shallower deep zones, animals emit light downward to match faint residual light from above, reducing their silhouette when viewed from below. At extreme trench depths, where sunlight is absent, this specific form becomes less relevant, but similar principles may apply relative to other light sources, such as bioluminescent organisms nearby. Some deep-sea shrimp and amphipods produce light not from internal organs, but through symbiotic bacteria. These bacteria live within specialized tissues, emitting light continuously. The host organism controls exposure through shutters or tissue movement. This partnership benefits both participants. The bacteria gain habitat and nutrients. The host gains illumination without synthesizing all components independently. Fish with photophores, specialized light-producing organs, often arrange them in species-specific patterns along their bodies. These patterns may function in recognition. Two individuals of the same species can identify each other by light arrangement even in total darkness. Researchers compare these arrangements to visual signatures, though understanding remains incomplete. The flashlight fish, though typically found in somewhat shallower waters, illustrates an interesting variation. It has light organs beneath its eyes containing symbiotic bacteria. The fish can rotate these organs, turning the light on or off from an observer's perspective. Similar control mechanisms likely exist among deeper species. In the deepest trench environments, where population density is low and encounters infrequent, energy-efficient signaling becomes valuable. Producing light constantly would consume unnecessary resources. Many species emit light only when stimulated by contact, chemical cues, or potential feeding opportunities. Bioluminescence also influences predator-prey dynamics indirectly. Predators capable of detecting faint glows gain advantage. Conversely, prey that remain non-luminous may avoid detection entirely. Some organisms evolved reduced or absent bioluminescent capacity specifically for concealment. Microbial communities contribute significantly to deep ocean light production. Certain bacteria glow steadily. When large aggregations colonize surfaces or host organisms, their combined light becomes visible even at a distance. Researchers studying sediment cores occasionally detect faint microbial luminescence under controlled conditions. The role of light in orientation remains an active research topic. Without sunlight, traditional visual navigation disappears. However, consistent bioluminescent background levels in some regions might provide orientation cues. Experiments continue, though replicating natural conditions in laboratories is challenging. Artificial lighting from research vehicles can temporarily disrupt natural light patterns. Animals sometimes approach out of curiosity or retreat depending on species. To reduce disturbance, scientists often use red illumination, which many deep-sea organisms cannot detect effectively. Evolutionary origins of bioluminescence appear multiple times independently across different lineages. Genetic studies show separate evolutionary pathways leading to similar functional outcomes. This suggests strong selective advantage in dark environments. When similar traits evolve repeatedly under comparable conditions, biologists often interpret it as convergent evolution. The energy cost of producing light remains relatively low compared to other biological processes, especially when reactions are efficient. Still, organisms regulate emission carefully. Light serves specific purposes rather than continuous illumination. Some squid species, though more common above trench depths, exhibit sophisticated light control, adjusting brightness, pulse rate, and even colour slightly. These abilities suggest neural regulation of photophores. Whether trench-dwelling squid possess comparable control remains under investigation. Human fascination with glowing marine life dates back centuries. Early sailors reported luminous seas caused by plankton blooms. Modern technology reveals deeper examples far removed from surface waters. The trench environment represents one of the least disturbed natural theatres for bioluminescence. Recording these lights requires sensitive cameras capable of detecting faint signals without overwhelming them with artificial illumination. Technological improvements in low-light imaging have expanded observation capabilities significantly in recent decades. Bioluminescence research also has practical applications. Luciferase enzymes are widely used in biomedical research as markers for gene expression. By linking luciferase genes to specific biological processes, scientists can track cellular activity through emitted light. This technique traces its origins partly to marine bioluminescent organisms. There are even investigations into sustainable lighting inspired by biological systems, though practical implementation remains complex. Biological light production is efficient, but requires living systems, making large-scale application challenging. In trench ecosystems, glowing organisms contribute to ecological balance. Predation, communication, mating, and camouflage all involve light. Remove bioluminescence, and behavioral dynamics would change significantly. Despite the importance of light production, many trench creatures remain entirely non-luminous. Transparency, chemical camouflage, and minimal movement provide alternative survival strategies. The ecosystem includes both glowing and non-glowing participants interacting continuously. The visual impression of a submersible drifting with lights off in deep water can be striking. Occasional flashes appear unpredictably. A brief glow moves past, then disappears. The darkness remains dominant, but it is punctuated by biological signals carrying information essential for survival. Each flash represents metabolic investment, evolutionary history, ecological interaction. It is not decorative, it is functional. In a world where sunlight never arrives, organisms generate their own illumination precisely when needed. As exploration continues, more species displaying unique luminescent capabilities are documented. Some produce sustained glows, Others emit rapid pulses. A few generate waves of light travelling along their bodies. The diversity suggests ongoing evolutionary experimentation within environmental constraints. And while attention gradually shifts toward other unusual trench inhabitants, including unexpectedly large crustaceans and organisms thriving on chemical energy rather than light, the presence of bioluminescent creatures remains a defining feature of deep ocean life, demonstrating that even in permanent darkness, visibility persists in brief, purposeful flashes shaped by survival needs rather than external illumination. As the lights from deep-sea exploration vehicles sweep slowly across the trench floor, one of the more surprising sights often appears not in the form of large fish or dramatic predators, but in creatures that, at first glance, resemble ordinary shrimp. Amphipods are crustaceans related to shrimp, crabs, and lobsters. In shallow coastal waters they are usually small, often just a few millimeters long, living among sand grains, algae, or rocks. They rarely draw much attention, yet in the Mariana Trench some amphipods grow far larger than most people would expect. The phenomenon is sometimes described informally as deep-sea gigantism. It does not mean every organism grows large at depth, but certain species do reach sizes noticeably greater than their shallow water relatives. Amphipods collected from trench depths have measured several centimetres long, occasionally more. That might not sound enormous compared to surface marine animals, but relative to their typical counterparts, it represents significant enlargement. Several factors contribute to this size difference. Temperature is one of the most important. Cold water slows metabolism. Growth occurs gradually, often over longer lifespans. When organisms grow slowly but live longer, they can reach larger eventual sizes. This pattern appears in multiple deep-sea species, not just amphipods. Pressure also plays a role indirectly. High pressure influences cellular processes, protein stability, and biochemical reaction rates. Species adapted to these conditions evolve metabolic strategies suited to slow, steady energy use rather than rapid bursts of activity. Over evolutionary time, body structures adjust accordingly. Food availability shapes amphipod behaviour strongly. In the trench, organic material reaching the seafloor is limited. Marine snow provides a constant but sparse input. Larger food falls, such as dead fish or marine mammals, are rare but important events. Amphipods act as scavengers. When a sizable carcass arrives, they can gather quickly in large numbers, consuming available tissue before other organisms arrive, or decomposition progresses too far. Researchers have documented swarms of amphipods around baited landers lowered into the trench. Within hours, sometimes minutes, dozens or hundreds may appear, their antennae probe constantly detecting chemical traces in the water. Chemoreception is highly developed in these animals. Vision plays a smaller role, especially in permanent darkness. Body structure reflects their scavenging lifestyle. Amphipods possess laterally compressed bodies, jointed legs, and specialized mouth parts adapted for grasping and tearing food. Their exoskeletons consist primarily of chitin reinforced with minerals. Some trench amphipods show incorporation of aluminum compounds into their exoskeletons, a feature rarely observed in shallow relatives. Scientists believe this may provide structural benefits under pressure. The research continues to clarify its exact function. Despite their somewhat larger size compared to shallow species, trench amphipods remain efficient energy users. Movement tends to be deliberate rather than rapid. They conserve energy whenever possible. When food appears, however, activity increases noticeably. Opportunistic feeding helps compensate for long intervals between substantial meals. Reproduction among deep amphipods follows crustacean patterns, but with environmental adjustments. Females typically brood eggs in a specialized pouch called a marsupium. Protecting developing embryos internally reduces exposure to harsh external conditions. Development times may be extended due to cold temperatures and slower metabolism. Population density in the trench varies. Some areas host relatively abundant amphipod communities, especially where organic matter accumulates. Other regions appear more sparsely populated. Currents, sediment type, and chemical composition all influence distribution. An interesting aspect of trench amphipods involves contaminant accumulation. Studies have detected persistent organic pollutants in specimens collected from extreme depths. Chemicals released into surface environments can travel long distances through ocean currents and sinking particles. The presence of such pollutants in one of Earth's deepest ecosystems highlights global environmental connectivity. From a physiological perspective, amphipods tolerate pressure through adaptations similar to other deep-sea organisms. Cellular membranes remain flexible. Proteins stabilize through biochemical mechanisms. Enzymatic activity continues despite compression. Unlike vertebrates, their exoskeletons do not rely on gas-filled spaces, reducing vulnerability to pressure-induced collapse. Their sensory systems emphasize chemical and tactile input. Long antennae extend forward, sampling the water continuously. Subtle vibrations transmitted through water and sediment provide additional cues. Eyes, when present, are often reduced or adapted specifically for detecting bioluminescence rather than ambient light. Bioluminescence itself occurs in some amphipod species, though not universally. Light production may serve defensive purposes or communication, but many trench amphipods rely primarily on chemical detection rather than visual signalling. Ecologically, amphipods play an essential recycling role. By consuming dead organic material, they help break down complex tissues into smaller components, accessible to microbes and other organisms. This contributes to nutrient cycling even in deep environments. Their presence also supports higher trophic levels. Some deep-sea fish prey on amphipods regularly. Snailfish, for example, have been filmed feeding on them near the trench floor. Thus amphipods occupy an intermediate position in the food web, linking organic input from above to larger predators. Research methods for studying trench amphipods include baited traps, remotely operated vehicle observations, and environmental DNA analysis. Each method provides partial insight. Direct observation reveals behaviour, traps allow anatomical study. Genetic sampling helps identify species diversity without requiring intact specimens. Pressure-retaining collection devices have improved specimen quality. Maintaining near-native pressure during ascent reduces tissue damage. This enables more accurate biochemical analysis and morphological examination. Laboratory studies show that some deep amphipods cannot survive long at surface pressure. Their physiology depends on compression. Decompression alters cellular function. This reinforces the idea that adaptation operates in both directions. What seems extreme to humans is normal to them. Comparisons with shallow amphipods reveal evolutionary divergence. Genetic studies suggest certain trench species descended from shallower ancestors, gradually adapting to deeper habitats. Over time, traits supporting pressure tolerance, energy efficiency, and chemical sensing became more pronounced. Size increase alone does not define trench amphipods. Structural reinforcement, metabolic adjustment, reproductive timing, and behavioral patterns all contribute. Gigantism represents just one visible aspect of a broader adaptive suite. There are still many unanswered questions. Exact lifespan remains uncertain for most species. Reproductive cycles in natural conditions are difficult to observe. Interactions with other trench organisms require more long-term monitoring. Technological advances continue to expand research capacity. Autonomous vehicles capable of extended deployments may provide continuous observation in the future. Improved imaging sensitivity could capture behaviour without disturbing natural conditions. Understanding amphipods contributes to broader ecological knowledge. Scavengers often indicate ecosystem health. Their abundance, contaminant load, and distribution patterns offer clues about organic input rates, pollution transport, and biological productivity even at extreme depths. Public perception sometimes focuses on dramatic deep-sea predators, but organisms like amphipods illustrate how ecosystems function quietly. They recycle nutrients, support food webs, and adapt steadily without drawing attention. Their larger-than-expected size in the trench environment reminds researchers that evolutionary outcomes depend heavily on environmental context. Temperature stability, pressure, energy scarcity, and absence of sunlight create conditions quite different from coastal habitats. As exploration continues deeper into trench ecology, amphipods remain a consistent presence. Cameras lowered thousands of meters often capture them first, moving across sediment, gathering near bait, or drifting slowly through the water column. Their familiarity contrasts with the strangeness of their surroundings. And while attention gradually shifts toward other trench inhabitants, including soft-bodied fish, luminescent organisms, and species adapted to chemical energy sources, the amphipods persist as reliable participants in this deep ecosystem, quietly demonstrating how life adjusts to extreme conditions through gradual, practical adaptations shaped over long periods beneath the constant weight of the ocean above. As cameras settle gently onto the trench floor and the sediment cloud begins to clear, one of the first vertebrates that may drift into view does not look imposing. It does not have armoured plates or exaggerated jaws. It does not glow brightly or dart quickly across the frame. Instead, it moves with slow precision, its body almost translucent, its outline soft against the darkness. This is the snailfish, one of the deepest living vertebrates ever recorded, and perhaps the clearest example that survival at extreme depth does not require bulk or aggression, but careful chemical and structural adaptation. The Mariana snailfish, often associated with the deepest zones of the Mariana Trench, has been documented at depths exceeding 8,000 meters. At those depths, pressure surpasses 800 times the pressure at sea level. The environment is near freezing, food is limited. Light does not exist except for brief flashes from other organisms, and yet this fish hovers calmly, moving as if these conditions are entirely ordinary. Physically, the snailfish appears delicate. Its body is soft and gelatinous, lacking the heavy musculature seen in fast-swimming shallow species. The skin is thin. In some specimens, internal organs are faintly visible through the body wall. There are no thick scales. The skeleton is lightly ossified, with reduced mineralization compared to surface fish. Cartilage replaces some of the denser bone structures found in related species living at shallower depths. That softness is not a weakness. It is a solution. At extreme pressure, rigid structures can become liabilities. Compression forces act uniformly from all directions. A flexible body distributes those forces more evenly, reducing stress points. Without large air-filled cavities such as swim bladders, the snailfish avoids the complications of gas compression entirely. Buoyancy is maintained through body composition rather than internal gas regulation. The tissues of deep snailfish contain high concentrations of trimethylamine N oxide, commonly abbreviated as TMAO. This compound stabilizes proteins under pressure, preventing them from unfolding or malfunctioning. Research has shown that TMAO levels in fish increase consistently with depth. In snailfish living at trench depths, concentrations are significantly higher than in shallow relatives. This biochemical adjustment allows cellular machinery to function despite extreme compression. However, TMAO concentration cannot increase indefinitely without consequences. It affects osmotic balance and water retention. Some researchers propose that vertebrate depth limits may partly reflect the upper threshold of TMAO practicality. The Mariana snailfish may be approaching that boundary, representing one of the deepest examples of vertebrate adaptation before biochemical constraints become prohibitive. Movement in snailfish reflects energy conservation. They do not sprint or chase prey over long distances. Instead, they hover just above the sediment, using subtle fin movements to maintain position. Their pectoral fins often appear broad relative to body size, assisting in stabilization rather than propulsion. The tail moves gently, enough to adjust orientation or approach prey. Diet primarily consists of small crustaceans, especially amphipods. Video footage from baited landers frequently shows snailfish arriving shortly after amphipods gather around food sources. Their feeding behaviour is deliberate. They position themselves carefully, then capture prey with quick but controlled mouth movements. There is little wasted motion. Their mouths are not dramatically oversized like some deep-sea predators, but they are effective for their ecological role. Teeth are small and suited for gripping rather than tearing large prey. This matches the available food supply in trench environments, where massive prey encounters are rare. Eyes in deep snailfish are functional but modest. Without sunlight, visual acuity does not require high resolution. Instead, detecting faint bioluminescent flashes becomes more important. Their visual systems are adapted accordingly, though research continues to clarify exact sensitivity ranges. Reproductive strategies remain partly uncertain due to observational challenges. Some related snailfish species lay egg masses attached to substrates, while others exhibit variations in brooding behaviour. At trench depths, sparse populations mean locating mates may depend on chemical signalling rather than visual cues. Chemical communication likely plays a central role in reproductive timing and partner identification. Growth rates are presumed slow, reflecting cold temperatures and limited energy availability. Metabolic processes operate at reduced pace. Longevity may therefore extend compared to similar-sized shallow fish, though definitive lifespan measurements remain difficult without long-term monitoring. One remarkable aspect of snailfish research involves the challenge of specimen retrieval. When brought rapidly to the surface, decompression can damage tissue severely. Maintaining near-native pressure during ascent requires specialized equipment. Even with careful handling, physiological changes may occur once pressure decreases. This complicates laboratory study and reinforces reliance on in-situ observation. Genetic analysis has provided insight into how snailfish evolved to inhabit extreme depths. Comparisons with shallower relatives reveal mutations affecting protein stability, membrane composition, and metabolic pathways. Over evolutionary time, populations likely migrated gradually into deeper habitats, accumulating adaptations with each incremental descent. The evolutionary path to extreme depth likely involved intermediate zones first. As certain individuals with slightly greater pressure tolerance survived deeper excursions, natural selection favoured those traits. Over generations, depth ranges extended further until distinct deep-dwelling species emerged. Interestingly, snailfish demonstrate that vertebrates can persist in trench ecosystems, where earlier scientific consensus predicted absence. For decades, it was widely assumed that vertebrate fish could not survive beyond certain depths. The documentation of snailfish in Challenger Deep revised that assumption significantly. Their presence also influences trench food web structure. As predators of amphipods and other invertebrates, snailfish help regulate scavenger populations. This creates balance within a system already constrained by limited energy input. Unlike dramatic portrayals of deep-sea monsters, snailfish do not appear threatening. Their gentle movements and soft bodies present a contrast to exaggerated imagery often associated with extreme environments. Yet their biological resilience may be among the most impressive traits observed in trench ecosystems. Temperature stability in the trench benefits their physiology. With minimal thermal fluctuation, regulatory systems can operate steadily. Combined with high pressure and darkness, this stability shapes consistent environmental parameters across generations. Comparative studies of snailfish from different depths reveal gradual morphological differences. Shallower species possess slightly more robust skeletons and less gelatinous tissue. As depth increases, bodies become progressively softer, bones lighter, and biochemical stabilizers more concentrated. This gradient illustrates evolution in action along environmental gradients. Rather than abrupt transformation, adaptation proceeds gradually, shaped by selective pressures specific to each depth zone. Snailfish also serve as ambassadors for understanding extreme vertebrate life. Public interest in their discovery has drawn attention to deep ocean exploration and the importance of continued research. Technological improvements in deep-sea imaging have enhanced observation quality. High-definition cameras capture subtle behaviours previously unseen. Long-duration landers allow extended monitoring, increasing data reliability. Despite progress, many questions remain. Exact population size estimates are uncertain. Distribution patterns across trench segments require further mapping. Seasonal variation, if any exists at such depths, remains under investigation. What is clear is that snailfish represent a biological limit case. They occupy one of the most extreme environments accessible to vertebrates on Earth. Their existence challenges assumptions about fragility and strength. Instead of heavy armour or aggressive speed, they rely on chemical stability, structural flexibility and energy efficiency. In the silent darkness of the trench floor under pressure capable of crushing most surface life instantly, the snailfish moves slowly but confidently. Its softness is not accidental. It is deliberate adaptation refined over countless generations. As exploration continues deeper into trench ecosystems and attention turns toward organisms thriving on chemical energy sources and species inhabiting hydrothermal gradients nearby, the snailfish remains a central example of how vertebrate life can extend further than once imagined, surviving not by resisting extreme conditions directly, but by integrating those conditions into every aspect of its biology, quietly embodying resilience in one of the most inhospitable places on the planet. As exploration moves farther across the trench floor and attention shifts from visible fish and crustaceans to the finer details of the environment, another layer of life begins to emerge. It does not swim past cameras in obvious shapes. It does not flash with bioluminescence or gather dramatically around bait. Instead, it exists quietly within sediments, on mineral surfaces, and sometimes inside other organisms. These are bacteria that thrive not on sunlight or conventional organic food sources, but on chemicals that many forms of surface life would find toxic. To understand how this works, it helps to remember that most ecosystems near the surface depend ultimately on sunlight. Plants, algae, and certain bacteria perform photosynthesis, converting light energy into chemical energy stored in organic molecules. Animals then consume those organisms or each other. Remove sunlight, and that familiar energy pathway disappears. Yet life in the deepest ocean persists. The alternative is chemosynthesis. Chemosynthesis is the process by which certain microorganisms generate organic matter, using chemical reactions rather than light. Instead of relying on solar energy, these bacteria extract energy from inorganic compounds such as hydrogen sulfide, methane, ammonia, or ferrous iron. Many of these chemicals are toxic to surface organisms at higher concentrations, but for specialized microbes, they represent fuel. In trench environments like the Mariana Trench, chemical gradients arise from several sources. Organic material settling from above decomposes, releasing compounds into the sediment. Geological activity associated with subduction zones contributes additional chemical flux. Hydrothermal activity is less dominant directly within Challenger Deep than along mid-ocean ridges, but localized seepage of reduced compounds still occurs. Hydrogen sulfide, for example, forms when organic matter decomposes in low oxygen conditions. To humans, it is associated with a strong odour and toxicity. Certain bacteria, however, oxidize hydrogen sulfide, using the released energy to fix carbon dioxide into organic molecules. These microbes form the base of localized food webs independent of sunlight. Methane provides another energy source. Methanotrophic bacteria oxidize methane, producing carbon dioxide and energy. Methane can originate from decomposing organic material buried in sediments or from geological processes deeper within Earth's crust. Again, What appears hazardous from one biological perspective becomes beneficial from another. Iron-oxidizing bacteria represent yet another group. They derive energy by converting ferrous iron to ferric iron. Such processes influence mineral deposition patterns and sediment chemistry. These microbial activities can leave visible signatures in sediment layers, detectable through chemical analysis. Ammonia-oxidizing bacteria participate in nitrogen cycling. They convert ammonia into nitrite and nitrate, processes essential for maintaining nutrient balance in marine ecosystems. Even in deep trenches, nitrogen cycling remains active, linking microbial metabolism to broader ocean chemistry. One particularly interesting aspect of these bacteria involves symbiotic relationships. Some larger deep-sea organisms host chemosynthetic bacteria within their tissues. Although more commonly documented around hydrothermal vents and cold seeps, similar associations may exist in trench environments. The host organism provides habitat and access to chemicals. The bacteria provide organic nutrients. This partnership allows animals to survive where conventional food sources are scarce. Tube worms near hydrothermal vents provide a well-known example outside the trench itself. They lack digestive systems entirely and rely on internal bacteria for nutrition. While trench equivalents are less dramatic, smaller-scale symbioses likely occur. Sediment cores taken from trench floors reveal dense microbial communities extending several centimetres or more below the surface. Oxygen penetration is limited, so anaerobic metabolic pathways dominate. Sulfate reduction, methane production, and other processes occur continuously. These reactions contribute to global carbon and sulfur cycles, even though they take place far from sunlight. Studying these microbes presents technical challenges. Sampling must avoid contamination from surface organisms. Pressure changes during ascent can alter microbial activity. Specialized sampling equipment helps preserve native conditions as much as possible. Genomic sequencing has become a powerful tool. Environmental DNA extracted directly from sediments reveals species diversity without requiring cultivation. Many deep-sea microbes resist traditional laboratory cultivation due to their specialized pressure and chemical requirements. Genetic methods allow identification and metabolic inference without full culture. Some trench bacteria exhibit extreme pressure tolerance known as piezophily. Their cellular membranes, enzyme systems, and genetic regulation function optimally under compression. At surface pressure, they may grow slowly or not at all. Their natural habitat defines their physiological baseline. Enzymes from these organisms interest biotechnology researchers. Pressure-stable enzymes may function under industrial conditions where conventional enzymes fail. Applications include food processing, pharmaceuticals, and environmental remediation. Certain deep-sea microbes also produce unique secondary metabolites. Some exhibit antimicrobial properties. Exploration of these compounds may contribute to antibiotic development, an area of ongoing research given global concerns about antibiotic resistance. The term toxic becomes relative in this context. Hydrogen sulfide, methane, and similar compounds are hazardous to humans at sufficient concentrations, but microbes capable of metabolizing them experience those chemicals as energy sources. Biological perspective determines whether a substance is harmful or useful. Energy yields from chemosynthesis are generally lower than from photosynthesis. Consequently, ecosystems based primarily on chemical energy tend to support lower biomass. Organisms grow slowly, populations remain relatively sparse, and energy conservation becomes critical. Even so, microbial activity forms the foundation for more complex trench life. By converting inorganic chemicals into organic matter, bacteria create food sources for other organisms, including small invertebrates. Those invertebrates in turn feed larger animals such as fish and crustaceans. The food web remains intact, though structured differently from surface systems. Chemical gradients in sediments also influence animal distribution. Some invertebrates prefer areas with higher microbial productivity. Others avoid zones with excessive sulfide concentrations. Microbial metabolism therefore shapes habitat suitability indirectly. Temperature stability in the trench benefits microbial communities. Near freezing conditions slow reaction rates, but remain consistent over time. Stability allows specialized metabolic pathways to persist without frequent environmental disruption. There is also growing interest in how these microbes contribute to long-term carbon storage. Organic carbon buried in deep-sea sediments can remain sequestered for geological timescales. Microbial processes influence whether carbon remains buried or returns to the water column as dissolved compounds. Planetary scientists study these systems as analogues for extraterrestrial environments. Subsurface oceans on icy moons may lack sunlight, but possess chemical energy sources. Understanding how Earth microbes exploit similar conditions informs hypotheses about potential life elsewhere. The presence of pollutants in trench sediments introduces another dimension. Persistent organic pollutants and microplastics have been detected alongside microbial communities. Some bacteria may metabolize certain contaminants, though research remains preliminary. Microbial mats occasionally form on sediment surfaces where chemical gradients are favourable. These mats may appear as thin films detectable under close observation. They represent concentrated metabolic activity zones. The resilience of these bacteria highlights a broader biological principle. Life adapts to available energy sources rather than requiring specific environmental features like sunlight. Given appropriate chemistry, evolution finds pathways for survival. Human perception often associates life with light, warmth, and oxygen-rich air. Trench microbes operate outside those familiar parameters. Their existence expands understanding of habitability. Ongoing research continues to identify new microbial lineages from trench samples. Many possess genetic sequences unlike previously documented organisms. This suggests substantial undiscovered diversity remains. Technological advances in deep-sea sampling, genomic sequencing, and chemical analysis promise further discoveries. Each expedition adds pieces to a complex ecological puzzle, and as exploration gradually shifts attention toward other trench inhabitants shaped by chemical gradients, including organisms clustering near seep zones and animals benefiting indirectly from microbial productivity, these bacteria remain foundational participants, quietly converting toxic chemicals into usable energy and sustaining life in one of Earth's most remote and chemically challenging environments, beneath kilometers of water where sunlight never reaches. As cameras drift slowly through the deep water above the trench floor, past amphipods and soft-bodied fish, another shape sometimes enters the frame. It does not glow gently like bioluminescent plankton. It does not hover delicately like a snailfish. Instead, it appears angular. Its head seems too large for its body. Its mouth opens wider than expected, and when the light catches it just right, rows of thin, needle-like teeth become visible, extending outward in a way that looks almost impractical. In the deep ocean, especially in zones above and along the trench slopes, predators have evolved jaws and teeth that appear exaggerated by surface standards. They are not oversized for drama. They are oversized because food is unpredictable. In sunlit waters, predators can afford selectivity. Prey may be abundant. Missed opportunities do not necessarily threaten survival. In the trench environment and its surrounding abyssal regions, meals may be separated by long intervals. When something edible appears, the safest strategy is to secure it immediately and efficiently, even if it is large relative to the predator's own body. One well-known example from deep waters is the fangtooth, a fish named for its disproportionately large teeth. Though not restricted solely to the Mariana Trench, fangtooth species inhabit deep ocean zones where similar pressures and darkness exist. Its teeth are so long that special pockets in the roof of its mouth accommodate them when the jaws close. Without those pockets, the teeth would pierce its own skull. The purpose of such teeth is straightforward. Slender, elongated fangs increase the probability of impaling slippery prey in darkness. Many deep-sea fish lack the ability to pursue prey over long distances. Instead, they rely on sudden encounters. When a potential meal comes within reach, sharp, protruding teeth ensure it cannot easily escape. The gulper eel offers another striking example. Its body is elongated and relatively thin, but its mouth expands dramatically. The lower jaw can hinge downward, creating a gape far larger than the head itself. Attached to the tail of some species is a small bioluminescent organ, possibly used to lure prey closer. When prey approaches, the eel can engulf it in a single rapid motion. The viperfish, often found in deep pelagic zones, possesses long, curved teeth that remain visible even when the mouth is closed. Its jaws can hinge widely, and it can swallow prey nearly its own size. The teeth are not designed for chewing, but for gripping. Once impaled, prey rarely escapes. Though these species are more common in the broader deep ocean than exclusively at Challenger Deep itself, similar anatomical principles apply in trench-adjacent ecosystems. The trench is not isolated from the rest of the deep sea. Water masses circulate slowly. Organisms may move between depth zones within their physiological limits. Jaw flexibility is essential in such environments. Ligaments connecting jaw bones in these predators are often elastic. Musculature is arranged to maximize opening width rather than crushing strength. Crushing requires energy and strong bone structures. Gripping and swallowing require less sustained force but more reach. Teeth composition also matters. Enamel and dentin structures must withstand pressure without becoming brittle. While pressure compresses uniformly, rapid jaw closure still generates localized mechanical stress. Teeth must balance hardness with resilience. Many of these predators exhibit relatively large heads compared to body size. The head houses not only jaws and teeth but sensory organs tuned to detect faint cues in darkness. Enlarged olfactory chambers allow detection of dissolved chemicals signaling nearby prey. Mechanosensory systems detect subtle water disturbances. Eyes vary depending on species. Some deep predators have relatively large eyes adapted for detecting bioluminescent flashes. Others have reduced vision but compensate through chemical and tactile sensing. Energy conservation influences this balance. Stomach expansion represents another adaptation. Elastic stomach walls allow ingestion of large prey items. In environments where feeding opportunities are rare, the ability to consume and store a substantial meal improves survival during long fasting intervals. Metabolic rates in deep predators are typically lower than in shallow water fish of comparable size. They do not chase prey continuously. Instead, they conserve energy until opportunity arises. When it does, their anatomical design ensures maximum capture efficiency. Reproductive strategies also reflect energy constraints. Some deep-sea predators exhibit sexual dimorphism. In certain anglerfish species, males attach permanently to females, fusing circulatory systems. While this specific behavior is more common above trench depths, it illustrates how extreme environments reshape reproductive biology. The appearance of these predators often captures public imagination. Large teeth, extended jaws, dark coloration. However, their size relative to surface predators remains modest. Most are not enormous creatures, but small to medium-sized fish with proportionally exaggerated features. Pressure tolerance remains foundational. Like other trench organisms, deep predators maintain high concentrations of stabilizing molecules such as TMAO. Their cellular membranes adjust lipid composition for flexibility under compression. Without these biochemical adaptations, their dramatic jaws would not function. Feeding behavior captured on deep cameras reveals patience rather than constant aggression. Predators often remain nearly motionless, conserving energy. When prey approaches within range, rapid jaw extension occurs. The strike is brief and decisive. Some species may use bioluminescent lures. Light production near the mouth can draw smaller organisms closer. In darkness, even faint glows attract attention. Once prey investigates, the predator's jaws close swiftly. The structure of the deep-sea food web limits predator abundance. With restricted energy input from above, biomass remains relatively low. Predators exist, but not in overwhelming numbers. Each individual must maximize feeding success. Comparisons with shallow water predators highlight environmental influence. Reef predators often display bright coloration and complex hunting strategies requiring maneuverability and speed. Deep predators emphasize stealth, energy conservation, and mechanical capture efficiency. Studying these species presents logistical challenges. Rapid decompression during retrieval can damage delicate tissues. Many specimens reach the surface distorted. Pressure-maintaining containers help but are not always feasible. High-definition in situ imaging has improved understanding of natural posture and movement. Earlier descriptions based on damaged specimens sometimes exaggerated proportions. Observing animals in their native pressure conditions reveals more accurate anatomy. Genetic analysis shows that many deep predators evolved from shallower ancestors gradually adapting to greater depths. Over evolutionary time, natural selection favoured individuals capable of seizing scarce prey effectively. There is also a boundary to vertebrate depth distribution. Below certain trench depths, vertebrate predators become rare or absent, possibly due to biochemical constraints. Invertebrate predators may extend deeper. Understanding this limit remains an active research topic. The phrase, impossible teeth and jaws, reflects surface perspective. Within trench conditions, these features represent practical solutions. When meals are uncertain and energy scarce, capturing prey decisively becomes critical. In darkness, there is no margin for error based on visual targeting. Mechanical efficiency compensates for limited sight. Long, curved teeth function like hooks. Wide jaws function like nets. Predators with such adaptations contribute to ecological balance. They regulate populations of amphipods, smaller fish, and other invertebrates. Even in low-energy systems, predator-prey dynamics shape community structure. As exploration continues, additional species with unusual jaw mechanisms may be documented. Each discovery refines understanding of evolutionary responses to extreme depth, and as focus gradually transitions toward other trench inhabitants, including organisms surviving without oxygen pockets or species exhibiting unexpected gigantism, these deep predators remain a striking reminder that in the blackness of the ocean's deepest regions, survival often depends not on strength alone, but on precision, efficiency, and anatomical specialization designed to secure rare opportunities in a world where nothing edible can be taken for granted. As exploration moves deeper across the trench floor and into the fine layers of sediment where larger fish rarely linger, another question naturally comes into focus. Oxygen, the gas most animals depend on for metabolism, is far less abundant in some deep ocean environments than near the surface. Circulation is slower. Organic material decomposes gradually, consuming available oxygen, and in certain pockets of sediment, oxygen may be almost entirely absent. Yet even here, life persists. For many animals living near the Mariana Trench, the challenge is not the complete absence of oxygen in the surrounding water column, but the scarcity of localized oxygen-rich microenvironments. Surface ecosystems benefit from constant atmospheric mixing. Waves, currents, and photosynthesis replenish oxygen continuously. In the deep ocean, especially near sediments where decomposition occurs, oxygen gradients can be steep. A few millimeters can separate relatively oxygenated water from nearly anoxic sediment layers. Some organisms avoid these zones entirely, remaining slightly above the sediment surface where oxygen levels remain higher. Others, however, interact directly with low oxygen environments. Certain invertebrates burrow into sediment for food or protection. Others feed on bacteria thriving in oxygen-poor layers. Survival under these conditions requires physiological adjustments. One key adaptation involves efficient oxygen extraction. Many deep-sea animals possess respiratory pigments with higher oxygen affinity than their shallow water counterparts. Hemoglobin variants, hemocyanin in some invertebrates, and other oxygen-binding molecules allow uptake even when dissolved oxygen concentration is low. These pigments bind oxygen more tightly, ensuring delivery to tissues despite scarcity. Metabolic rate reduction represents another strategy. Lower metabolic demand reduces oxygen consumption. Cold temperatures in trench environments naturally slow biochemical reactions, supporting this adaptation. Animals conserve energy, move slowly, and limit unnecessary activity. This approach aligns with the overall low-energy character of deep ecosystems. Certain worms, mollusks, and crustaceans inhabiting trench sediments exhibit tolerance for intermittent hypoxia, periods of low oxygen availability. They may switch temporarily to anaerobic metabolic pathways, producing energy without oxygen. This process generates metabolic byproducts requiring later clearance when oxygen becomes available again. The ability to shift between aerobic and anaerobic metabolism increases survival flexibility. Some animals maintain symbiotic relationships with bacteria capable of anaerobic metabolism. These bacteria process chemicals within the host organism or in its immediate environment. In exchange, they provide organic nutrients. While more extensively documented around hydrothermal vents and cold seeps, similar interactions may occur in trench sediments. There are also species that position themselves strategically at oxygen gradients. For example, certain small crustaceans and worms remain partially buried in sediment, with respiratory structures extending into more oxygenated water. This positioning allows access to both food sources within sediment and oxygen from overlying water. Sediment chemistry plays a major role. As organic material decomposes, microbes consume oxygen first. Once oxygen is depleted, other chemical pathways dominate, including sulfate reduction and methane production. Animals inhabiting these zones must tolerate associated chemical changes, including elevated hydrogen sulfide concentrations, which can be toxic to many organisms. Some invertebrates possess detoxification mechanisms, enabling survival in sulfide-rich environments. Specialized proteins bind sulfide molecules, reducing their toxicity. Others maintain behavioral avoidance, retreating temporarily when concentrations rise. It is important to clarify that most multicellular animals in the Mariana Trench still rely on oxygen to some degree. Completely oxygen-independent animal metabolism remains extremely rare and mostly documented in isolated cases elsewhere, such as certain microscopic organisms. However, trench animals often function effectively at oxygen concentrations much lower than typical surface levels. Fish inhabiting trench depths, including snailfish, rely on dissolved oxygen in surrounding water. Their gill structures, blood chemistry, and circulation systems adapt for efficient extraction. Slow swimming and reduced muscle mass decrease oxygen demand. Invertebrates such as amphipods exhibit similar strategies. Their exoskeletons house gill-like structures that maximize surface area for oxygen uptake. Movement patterns minimize unnecessary energy expenditure. One interesting aspect involves the interface between oxygenated water and anoxic sediment. This boundary hosts active microbial communities. Animals feeding at this interface benefit from microbial productivity, while still accessing sufficient oxygen slightly above the sediment. Temperature stability again influences oxygen dynamics. Cold water holds more dissolved oxygen than warm water, partially compensating for slow circulation. However, localized depletion still occurs due to decomposition processes. Researchers studying oxygen dynamics in the trench use specialized sensors deployed on landers and remotely operated vehicles. These sensors measure dissolved oxygen concentration at fine spatial resolution. Results reveal complex gradients rather than uniform conditions. Human influence appears even here. Climate change affects global ocean circulation patterns, potentially altering oxygen distribution. Deoxygenation trends observed in some ocean regions may extend gradually to deeper waters over long timescales. Monitoring trench oxygen levels contributes to understanding these broader processes. Another factor involves bioturbation, the disturbance of sediment by organisms. Burrowing animals mix sediment layers, influencing oxygen penetration. Even small organisms contribute to sediment aeration indirectly. Some deep-sea organisms exhibit periodic vertical movement. By shifting position relative to oxygen gradients, they optimize respiration and feeding simultaneously. These movements are usually slow and subtle. Laboratory experiments simulating low oxygen conditions help clarify tolerance limits. Maintaining pressure and temperature conditions simultaneously remains technically challenging, but controlled studies provide useful insights into respiratory physiology. Evolutionary adaptation to hypoxia likely occurred gradually. Populations exposed to slightly lower oxygen concentrations over generations accumulated beneficial traits. These traits include enhanced respiratory pigments, metabolic flexibility, and behavioral adjustments. Genomic studies identify genes associated with hypoxia tolerance. Expression patterns indicate upregulation of pathways supporting anaerobic metabolism, oxidative stress resistance, and efficient oxygen transport. Understanding these adaptations has broader implications. Medical research into hypoxia tolerance, such as improving outcomes for patients experiencing low oxygen conditions, sometimes draws inspiration from naturally adapted organisms. Energy conservation remains central. Without abundant oxygen, aerobic metabolism efficiency decreases. Organisms compensate by minimizing expenditure rather than increasing intake. Despite low oxygen availability in certain zones, trench ecosystems maintain functional food webs. Microbes process organic matter. Invertebrates consume microbes. Fish prey on invertebrates. Each level adapts physiologically to prevailing oxygen conditions. Observational footage often shows animals maintaining position just above sediment, suggesting careful balance between oxygen access and feeding opportunity. Rarely do they burrow deeply unless specifically adapted for it. In the broader context of extreme environment biology, trench organisms illustrate how life tolerates multiple simultaneous stresses: pressure, darkness, cold, chemical gradients, and limited oxygen. Adaptation rarely addresses a single factor in isolation. As exploration continues and attention gradually shifts toward phenomena such as deep-sea gigantism and hydrothermal vent ecosystems influencing trench-adjacent areas, the presence of animals capable of functioning under reduced oxygen availability remains another example of evolutionary flexibility, demonstrating that even in environments where oxygen pockets are scarce and chemical conditions fluctuate, life persists through physiological efficiency, metabolic flexibility, and behavioral precision shaped steadily over long periods beneath the surface of the deepest ocean. As the slow exploration of trench ecosystems continues, one pattern becomes increasingly noticeable. While many deep-sea organisms appear delicate, translucent, or reduced in size to conserve energy, some species show the opposite trend. Certain crustaceans, worms, and other invertebrates grow significantly larger than their relatives in shallower waters. The effect is not universal, but it is consistent enough that scientists refer to it as deep-sea gigantism. The reasons behind it are complex and still actively studied, yet several factors combine to make unusually large body size a workable strategy in the abyss. Temperature plays a major role. The deep ocean remains cold year-round, typically just above freezing. Cold conditions slow metabolic processes. Growth becomes gradual. When organisms grow slowly but live longer, they often reach larger eventual sizes. This pattern appears in multiple environments beyond the trench, including polar seas, which share similar temperature stability. Slow metabolism reduces wear on tissues, allowing extended lifespans and continued incremental growth. Longevity alone does not guarantee large size. but it creates the possibility. When food intake remains sufficient, even if intermittent, extended lifespan allows cumulative growth that surpasses what shallow relatives achieve in shorter lifetimes. Amphipods in the Mariana Trench illustrate this pattern clearly. Some species grow several times larger than coastal amphipods, not because they grow faster, but because they grow steadily over longer periods. Oxygen dynamics also influence size. Cold water holds more dissolved oxygen than warm water. Even though deep circulation is slower, the temperature factor partially compensates. Larger body size can be supported when oxygen supply meets metabolic demand. This relationship between oxygen availability and body size has been observed in various marine organisms. Pressure itself contributes indirectly. Under extreme compression, physiological processes adjust towards stability rather than rapid activity. Animals that move slowly and conserve energy can maintain larger bodies without proportional increases in metabolic expenditure. Flexible tissues distribute pressure evenly, reducing structural strain even in larger organisms. Food distribution patterns encourage opportunistic growth. In trench environments, large food falls are rare but significant. A whale carcass, for example, can support a community of scavengers for months or years. Organisms capable of consuming and storing substantial energy from such events gain survival advantages. Larger body size enables storage of nutrients in tissues, extending survival between feeding opportunities. Predation pressure differs from surface ecosystems. Fewer visually hunting predators operate in total darkness. While predation still occurs, certain protective advantages of large size, such as deterrence or reduced vulnerability, may outweigh the energetic costs. For some invertebrates, increased size reduces the number of potential predators capable of handling them. Reproductive strategies also connect to size. Larger individuals often produce more eggs or larger offspring, improving reproductive success in environments where encounters between mates are infrequent. Extended lifespan increases the number of reproductive cycles possible over time. Examples of abyssal gigantism include not only amphipods but also isopods, sea spiders, certain squid species, and some deep-sea worms. Giant isopods, for instance, resemble oversized pill bugs, reaching lengths far greater than terrestrial relatives. Though typically associated with abyssal plains rather than the deepest trench floor itself, they illustrate how similar environmental pressures produce comparable outcomes. Sea spiders in deep waters can grow significantly larger than shallow counterparts. Their elongated limbs allow efficient movement across soft sediments, while maintaining low metabolic expenditure. The spacious body cavity accommodates reproductive structures and digestive extensions. Squid species inhabiting deeper waters sometimes achieve impressive size, though their distribution varies. Reduced predation and stable temperatures may contribute to this trend. Documentation remains incomplete due to observational difficulty at depth. Gigantism does not occur uniformly across all taxa. Some species remain small because small size offers advantages such as lower energy requirements, faster reproduction, or easier concealment. Evolution balances multiple pressures rather than favouring size alone. Energy efficiency remains critical. Large deep-sea organisms often exhibit slow movement patterns. Musculature emphasises endurance over speed. Behavioural strategies include extended periods of inactivity punctuated by feeding or reproductive activity. Structural adaptations accompany increased size. Exoskeletons in large crustaceans maintain flexibility under pressure. Soft tissues distribute compressive forces. Calcified structures, when present, often incorporate biochemical stabilizers ensuring resilience. Genetic studies suggest that gigantism evolves gradually through cumulative selection. Individuals slightly larger than average may gain survival advantages under certain conditions. Over generations, average size shifts upward. There is also discussion among researchers about the island rule analogy applied to deep seas. On islands, Small species sometimes grow larger due to reduced predation and competition. Similarly, isolated deep-sea habitats may encourage size increase in certain taxa. Environmental stability contributes as well. Surface ecosystems experience seasonal changes, storms, temperature fluctuations, and varying food supply. The deep ocean, particularly trench environments, remains comparatively stable. Stability allows long-term growth strategies without frequent environmental resets. Another factor involves developmental timing. Slower development under cold conditions may alter hormonal regulation of growth. Extended juvenile phases can lead to larger adult size once maturity occurs. Gigantism may also enhance sensory capabilities. Larger antennae in crustaceans increase chemical detection range. Extended limbs improve substrate exploration. Larger body mass supports more developed sensory organs. Scientific investigation into abyssal gigantism benefits from deep-sea imaging technology, pressure-retaining sampling devices, and genomic analysis. Each method contributes to understanding how environmental variables translate into biological outcomes. Public fascination often focuses on dramatic examples like giant squid, though those species inhabit a range of deep pelagic zones rather than trench floors exclusively. Still, the principle remains similar. Stable, cold environments and reduced predation can favour larger body sizes. Gigantism also intersects with ecosystem function. Larger scavengers process organic matter differently than smaller ones. Their feeding activity influences sediment structure, nutrient distribution, and microbial activity. Long lifespans associated with larger size may make these species sensitive to environmental change. Pollution, climate-driven oxygen shifts, or altered organic input could impact populations gradually but significantly. Researchers continue to examine how climate change might influence deep-sea gigantism. Warming oceans could alter circulation patterns, oxygen availability, and organic matter deposition, potentially affecting size trends over long timescales. From a human perspective, encountering unexpectedly large crustaceans or invertebrates in deep footage creates a sense of unfamiliar scale. Creatures resembling familiar shoreline species appear enlarged, emphasizing how environment shapes morphology. Despite increased size, these organisms rarely exhibit aggressive behavior toward exploration equipment. Their movements remain deliberate, consistent with energy conservation strategies. Understanding gigantism provides insight into evolutionary flexibility. Life does not follow a single trajectory toward either smaller or larger forms. Instead, size reflects a balance among temperature, energy supply, predation, reproduction, and physiology. As attention continues shifting toward other aspects of trench ecology, including hydrothermal influences and ecosystems independent of sunlight, the presence of unusually large organisms remains another example of how extreme environments reshape biological expectations, demonstrating that in the quiet stability of the abyss, slow growth combined with longevity can produce creatures larger than anticipated, adapted not for speed or display, but for persistence in one of Earth's most stable yet demanding habitats beneath the ocean surface. As the exploration of the trench ecosystem widens beyond the flat sediment plains and into areas shaped by tectonic activity, another environment comes into view. It is not defined only by pressure and darkness, but by heat rising from deep within the Earth. Along subduction zones and fault lines near the Mariana region, hydrothermal vents release mineral-rich fluids into the surrounding water. These vents create localized ecosystems that operate under rules very different from those governing the rest of the trench floor. Hydrothermal vents form where seawater seeps into cracks in the ocean crust, travels downward, and becomes superheated by underlying magma. The water reacts chemically with surrounding rock, dissolving minerals and absorbing reduced compounds such as hydrogen sulfide, methane, iron, and manganese. Eventually, this heated, chemically altered water rises back to the seafloor and emerges through vent openings. When it meets the cold, deep ocean water, minerals precipitate, forming chimney-like structures that can grow several meters tall. In shallow waters, heat and chemical plumes might disperse quickly. In the deep ocean, especially in relatively stable trench environments, vent systems can persist for long periods. Around them, life organises itself in dense clusters. The contrast between the barren surrounding sediment and the concentrated activity near a vent can be striking. Unlike surface ecosystems that depend on sunlight, vent ecosystems rely on chemosynthesis. Specialized bacteria oxidize chemicals such as hydrogen sulfide, using the released energy to fix carbon dioxide into organic matter. These bacteria form the foundation of the vent food web. Larger organisms either feed directly on them or host them symbiotically. One of the most recognizable vent inhabitants is the giant tube worm, particularly species such as Riftia pachyptila. Though these are more common at mid-ocean ridges than directly within the deepest trench floor, similar principles apply in Mariana region vent systems. Tube worms lack mouths and digestive tracts. Instead, they contain an internal organ called a trophosome, filled with chemosynthetic bacteria. The worms absorb hydrogen sulfide, oxygen and carbon dioxide from the surrounding water. The bacteria convert these into nutrients, effectively feeding the host. Crabs and shrimp often gather around vents as well. Some species possess specialized heat-tolerant proteins, allowing them to survive near vent openings, where water temperatures can exceed 300 degrees Celsius before mixing with cold seawater. Although the immediate vent fluid is extremely hot, rapid mixing creates steep temperature gradients. Animals position themselves carefully within tolerable ranges. Vent shrimp sometimes exhibit reduced eyesight or modified eyes that detect infrared radiation or faint glows from vent emissions. Traditional vision is less useful in darkness, but detecting heat signatures or chemical gradients can guide positioning. Snails and mussels form dense colonies near certain vents. Many of these organisms also host chemosynthetic bacteria within their tissues. Symbiosis allows them to access nutrients without relying solely on free-living bacterial mats. The mineral composition of vent fluids influences which organisms thrive. Sulfide-rich vents support sulfur-oxidizing bacteria. Methane seeps support methanotrophic bacteria. Each chemical environment fosters slightly different biological communities. Pressure at vent sites remains extreme, especially in deep trench regions. Organisms must tolerate both compression and chemical exposure. Heat adds another stress factor. Proteins in vent animals often possess enhanced stability to withstand fluctuating temperatures. Heat shock proteins help maintain cellular integrity under thermal stress. Vent ecosystems are often described as oases in the deep sea. The surrounding trench floor may host sparse life dependent on marine snow, while vent areas exhibit concentrated biomass supported by chemical energy. This contrast highlights how energy availability shapes biological density. Geological activity influences vent lifespan. Some vents remain active for decades or centuries. Others shut down as tectonic conditions change. When a vent ceases activity, dependent communities must disperse or perish. Larval stages of many vent species drift through deep water, seeking new active sites. The discovery of hydrothermal vents in the late 1970s fundamentally altered understanding of deep-sea biology. Prior to that, it was assumed that life without sunlight would be sparse and simple. Vent ecosystems demonstrated that complex, densely populated communities could exist independent of solar energy. In the Mariana region, hydrothermal systems may be less extensive than along major mid-ocean ridges, but subduction zones still generate chemical seepage capable of supporting localized chemosynthetic life. Exploration continues to identify additional vent fields. Sediment composition around vents differs from surrounding areas. Mineral deposits create hard substrates for attachment. Chimney structures accumulate layers of metal sulfides, forming striking formations in otherwise flat terrain. Vent organisms often display rapid growth relative to other deep-sea species due to concentrated energy availability. Compared to the slow metabolic pace of trench floor scavengers, vent communities may appear comparatively active. Reproductive strategies vary. Some species release larvae into water currents, relying on dispersal to colonize distant vents. Others maintain localized reproduction near active sites. Genetic studies reveal connectivity between vent populations separated by significant distances, suggesting occasional long-range dispersal events. The chemistry of vent fluids affects global ocean cycles. Elements released from Earth's crust contribute to seawater composition. Microbial metabolism near vents influences carbon and sulfur cycling on planetary scales. Research into vent life also informs astrobiology. Subsurface oceans beneath icy moons may host hydrothermal activity driven by tidal heating. If chemical gradients similar to Earth's vents exist elsewhere, they could support microbial ecosystems independent of sunlight. Technological challenges in studying vents include extreme temperature gradients, high pressure, and corrosive chemical conditions. Remotely operated vehicles equipped with heat-resistant materials and chemical sensors enable sampling. Scientists analyze vent fluids for mineral content, microbial diversity, and isotopic signatures. These data help reconstruct geological and biological interactions. One fascinating observation involves vent animals tolerating rapid environmental change. As vent output fluctuates, organisms reposition themselves along temperature and chemical gradients. Behavioral flexibility complements physiological adaptation. Vent ecosystems illustrate that toxicity is relative. Hydrogen sulfide, lethal to many surface animals, becomes primary energy source for bacteria. Animals hosting these bacteria convert toxicity into nutrition through partnership. Despite the dense activity near vents, surrounding trench areas remain relatively sparse. This patchy distribution creates ecological contrast within short distances. Vent chimneys can collapse or become buried by sediment over time. When this happens, communities shift or relocate. Succession patterns resemble ecological changes in terrestrial environments following disturbance. Ongoing exploration aims to quantify biodiversity at Mariana region vents more precisely. Many species likely remain undocumented due to limited survey coverage. While hydrothermal vents are not uniformly distributed across the trench floor, their presence adds complexity to trench ecosystems. They demonstrate that even in the deepest, darkest parts of the ocean, internal planetary heat drives localized life-supporting systems. And as attention gradually transitions from these chemically fueled communities toward broader questions about evolutionary isolation and undiscovered species still waiting in unexplored depths, the ecosystems surrounding hydrothermal vents stand as clear evidence that life does not require sunlight to flourish. Given chemical energy, stable pressure conditions, and time, organisms can assemble intricate self-sustaining communities in places that, from a surface perspective, might seem entirely incompatible with dense biological activity. As exploration continues deeper into the trench environment and attention moves beyond individual species toward the broader functioning of the ecosystem, one fundamental difference between surface life and deep ocean life becomes increasingly clear. Sunlight, which powers almost every familiar food chain on land and in shallow seas, plays no role here. There is no photosynthesis on the trench floor, no plants, no algae forests, no sunlight-driven plankton blooms, and yet life persists in organized, interconnected systems. To understand survival in this environment, it helps to think in terms of energy pathways rather than specific organisms. At the surface, energy enters ecosystems primarily through photosynthesis. Plants convert light into chemical energy. Herbivores eat plants. Carnivores eat herbivores. The structure is familiar. In the deep ocean, particularly in trench environments where light never penetrates, that initial step cannot occur locally. energy must arrive through alternative means. One of the primary sources is organic matter sinking from above. Tiny fragments of plankton, microscopic algae, animal waste and dead organisms drift downward continuously. This slow, steady fall is often called marine snow because of its visual resemblance to snowflakes descending through water. It may seem insignificant particle by particle, but over time it supplies essential nutrients. The amount of marine snow reaching trench depths is limited. Much of it is consumed or decomposed long before it arrives. What remains supports a sparse but persistent food web. Small invertebrates feed directly on these particles. Larger scavengers feed on those invertebrates. Fish such as snailfish prey on crustaceans like amphipods. The chain continues upward, even in the absence of sunlight. Occasionally, larger food falls dramatically increase available energy. A dead whale, large fish, or other sizable carcass sinking into the trench can sustain a community for extended periods. These events are rare but important. Scavengers gather quickly, microbes begin decomposition, and nutrients spread into surrounding sediment. Such pulses of organic input influence population dynamics and reproductive cycles. Chemosynthesis provides another pathway entirely independent of sunlight. Certain bacteria derive energy from inorganic chemicals, converting compounds like hydrogen sulfide, methane or ammonia into organic matter. These microbes form the base of localized ecosystems, particularly near hydrothermal vents or chemical seep zones. Animals either consume these bacteria directly or host them symbiotically. The existence of chemosynthetic ecosystems significantly expanded scientific understanding when first discovered. Before deep-sea vent exploration, it was widely assumed that sunlight was an absolute requirement for complex ecosystems. Chemosynthesis demonstrated that chemical energy alone could support organized biological communities. Microbial decomposition also plays a central role. Organic material reaching the trench does not remain intact indefinitely. Bacteria break it down, releasing nutrients back into the system. This recycling sustains invertebrates feeding within sediments and supports broader nutrient cycling. Sediment layers themselves act as nutrient reservoirs. Organic particles accumulate over time. Burrowing organisms disturb these layers, mixing nutrients and oxygen. This process, known as bioturbation, influences microbial activity and nutrient availability. Energy conservation becomes a defining survival strategy in the absence of photosynthesis. Organisms tend to move slowly, reduce unnecessary activity, and maintain efficient metabolic pathways. Rapid growth and frequent reproduction, common in sunlit ecosystems, occur less often in trench environments. Temperature stability reinforces this pattern. Cold water slows metabolism naturally. Organisms adapted to such conditions often exhibit extended lifespans, slow maturation, and infrequent reproductive cycles. Energy expenditure aligns closely with limited intake. Body composition reflects energy economics. Gelatinous tissues require less structural investment than dense muscle or heavy skeletal material. Many deep-sea organisms display soft bodies, reduced bone density, and minimal pigmentation. These features reduce metabolic cost. Sensory adaptations also reflect energy considerations. Complex visual systems require substantial neural investment. In permanent darkness, chemical sensing and mechanoreception provide more efficient alternatives. Long antennae, sensitive lateral lines, and enhanced olfactory structures allow environmental awareness without heavy visual processing. Reproductive strategies adjust accordingly. Some species produce fewer but more developed offspring. Others rely on chemical cues to locate mates over distances. Hermaphroditism occurs in certain deep-sea organisms, increasing reproductive flexibility when encounters are rare. Microbial partnerships enhance survival further. Symbiotic bacteria living within animal tissues provide nutritional supplementation. In exchange, the host supplies habitat and chemical substrates. These relationships blur traditional distinctions between producer and consumer. The absence of photosynthesis also influences carbon cycling. Organic carbon reaching the trench often becomes buried in sediments. Some remain sequestered for geological timescales. Microbial processes determine how much carbon is recycled versus stored. Global ocean circulation connects surface productivity to deep ecosystems. Regions with high plankton productivity contribute more marine snow. Seasonal variations at the surface may subtly influence deep food supply, though time delays occur due to slow sinking rates. Human activity indirectly affects trench ecosystems. Changes in surface productivity, climate-driven shifts in ocean circulation, and pollution transport all influence energy input. Even remote deep environments remain linked to surface processes. Research methods for studying non-photosynthetic ecosystems include sediment core analysis, isotopic tracing, and environmental DNA sampling. These approaches reveal nutrient sources, metabolic pathways, and species diversity without relying solely on visual observation. Stable isotope analysis helps identify energy origins. Carbon and nitrogen isotopic ratios indicate whether organisms derive nutrition from surface organic matter or chemosynthetic sources. Such analyses clarify food web structure. Technological advances in deep-sea landers allow long-term monitoring. Time-lapse imaging reveals scavenger arrival patterns, decomposition rates, and community succession following large food falls. The concept of survival without photosynthesis also informs astrobiology. Many planetary bodies lack surface sunlight but may possess subsurface oceans with chemical energy sources. Studying Earth's deep ocean provides models for potential extraterrestrial ecosystems. Evolutionary implications are significant. Life likely began in environments without photosynthesis billions of years ago. Chemosynthetic pathways may represent ancient metabolic strategies predating oxygen-rich atmospheres. Modern trench ecosystems offer glimpses into early Earth conditions. Educationally, understanding these systems challenges assumptions about life requirements. Light, warmth, and abundant oxygen characterize human environments, but are not universal prerequisites for biological complexity. Energy limitation shapes ecological interactions. Competition occurs, but intensity differs from nutrient-rich environments. Cooperation through symbiosis often becomes advantageous. Despite energy scarcity, biodiversity persists. Microbial diversity is particularly high. Many species remain undiscovered due to sampling challenges and limited exploration coverage. As research expands, scientists continue identifying novel metabolic pathways, unusual symbiotic relationships, and previously undocumented organisms adapted specifically to life without sunlight. And as attention gradually shifts toward how isolation influences evolutionary divergence in trench species, and the possibility of undiscovered organisms still inhabiting unexplored depths, the ability of life to persist without photosynthesis remains a central theme, demonstrating that given chemical energy, stable environmental conditions, and sufficient time, ecosystems can develop and maintain complexity even in complete and permanent darkness beneath the deepest waters of the ocean. As the quiet landscape of the Mariana Trench stretches outward beyond the reach of submersible lights, another defining feature of this environment becomes clear. It is not only deep, dark, cold, and pressurized. It is also profoundly isolated. Unlike coastal seas where currents mix populations regularly, or shallow reefs where migration is relatively easy, trench ecosystems are separated by depth barriers, temperature layers, pressure gradients, and vast horizontal distances. This isolation shapes evolution in slow but very distinctive ways. Isolation in evolutionary terms usually means limited gene flow between populations. When groups of organisms cannot easily interbreed with others, genetic differences accumulate generation after generation. Mutations that might be diluted in large interconnected populations instead persist locally. Over time, these differences can produce entirely new species, adapted specifically to their confined environment. The Mariana Trench represents one of the most extreme examples of such separation on Earth. The depth itself acts as a barrier. Many organisms capable of living in shallower waters cannot tolerate the pressure at trench depths. Conversely, species adapted to extreme pressure often cannot survive if they move upward, where compression decreases. This vertical separation functions much like geographic isolation on land. Temperature stability reinforces this boundary. Surface waters fluctuate seasonally. The trench remains near freezing consistently. Organisms adapted to stable cold conditions may struggle with temperature variation elsewhere. Again, movement becomes limited and isolation persists. Food availability also shapes evolutionary pathways. Limited energy input means population sizes tend to remain relatively small. Smaller populations can experience genetic drift more strongly than large populations. Traits may spread or disappear partly due to chance rather than direct selection pressure. Over long timescales, drift contributes to distinct evolutionary outcomes. Examples of trench-specific species illustrate this process. The Mariana snailfish represents a lineage that appears to have adapted progressively to deeper environments. Genetic studies suggest ancestors lived at shallower depths. Gradual adaptation to increasing pressure, darkness, and limited food availability eventually produce species specialized for trench life. Amphipods collected from different trench systems often show genetic differences even when they appear similar morphologically. Separate trenches function like isolated basins. Organisms rarely migrate between them because crossing shallower, warmer, lower pressure regions may exceed physiological tolerance. Microbial communities also reflect isolation. Sediment DNA analysis reveals unique bacterial lineages not commonly found elsewhere. Chemical gradients, pressure stability, and local mineral composition create distinct ecological niches. Hydrothermal vent communities demonstrate similar patterns. Vent systems separated by distance often host related but distinct species. Larval dispersal occurs, but successful colonization depends on finding compatible chemical conditions. Many larvae likely fail to establish populations, reinforcing isolation. Time plays a critical role. Evolution in extreme environments often proceeds slowly due to reduced metabolic rates and long generation times. Cold temperatures and energy scarcity extend lifespans and delay reproduction. While evolutionary change still occurs, it may unfold over extended geological periods. Isolation sometimes produces unexpected morphological traits. Reduced eyes in permanent darkness, softened skeletons under pressure, enhanced chemical sensing capabilities. These traits reflect selective pressures unique to trench conditions rather than universal evolutionary trends. Behavioral adaptations evolve as well. Energy conservation strategies, slow movement patterns, opportunistic feeding, and infrequent reproduction characterize many trench species. These behaviors become genetically reinforced over generations. Isolation can also limit competition. Fewer species reach trench depths compared to shallow ecosystems. Reduced competition allows specialized species to occupy ecological roles without constant displacement. However, it also means disturbances may have significant impact because replacement populations are limited. Predator-prey relationships adapt accordingly. Predators develop efficient capture mechanisms suited to scarce prey encounters. Prey evolve camouflage, transparency, or chemical defenses appropriate for dark, high-pressure conditions. Chemical communication becomes especially important in isolated environments. Sparse populations require reliable signaling methods for reproduction and territorial interactions. Pheromone sensitivity often increases accordingly. Genomic research has begun revealing molecular signatures of trench adaptation. Genes related to protein stability, membrane flexibility, hypoxia tolerance, and metabolic efficiency show distinctive expression patterns. Comparative genomics helps identify evolutionary pathways leading to extreme environment specialization. Another interesting aspect involves convergent evolution. Separate trenches host organisms with similar adaptations despite independent evolutionary histories. Soft bodies, reduced pigmentation, enhanced chemosensory structures appear repeatedly. Environmental constraints shape similar solutions. Isolation does not mean complete stagnation. Occasional disturbances introduce change. Seismic events can redistribute sediment. Large carcass falls alter nutrient availability. Ocean circulation patterns shift over long timescales. Each event introduces selective pressure influencing evolutionary direction. Climate change introduces additional variables. Surface productivity affects marine snow input. Oxygen distribution patterns may shift gradually. Although deep environments buffer short-term change, long-term trends still influence trench ecosystems indirectly. There is also a philosophical dimension to evolutionary isolation. Surface ecosystems are dynamic, interconnected, visibly active. The trench represents the opposite. Change occurs, but quietly, slowly, often invisibly without specialized instruments. Human exploration itself represents a new factor. Sampling, noise, pollution transport, and climate-driven ocean changes introduce influences previously absent. Monitoring ensures minimal disruption, but awareness of potential impact remains important. Studying evolution in extreme isolation contributes to broader biological understanding. It helps clarify how life adapts when migration options are limited, and environmental conditions remain stable over long periods. These insights apply beyond Earth as well. Planetary scientists consider isolated subsurface oceans on icy moons potential habitats for life. If organisms exist there, they may evolve under similar isolation conditions. Isolation also shapes biodiversity patterns. Some trenches may harbour endemic species found nowhere else. Documentation remains incomplete due to limited exploration coverage. Technological advances such as environmental DNA sampling, autonomous vehicles and improved deep-sea imaging continue expanding knowledge. Each expedition reveals new species or new aspects of known organisms. Understanding evolutionary isolation helps interpret morphological and physiological traits accurately. What appears unusual from a surface perspective often reflects logical adaptation within a constrained environment. Energy limitation, pressure stability, darkness, and chemical gradients combine to produce unique evolutionary pathways. None of these factors operate alone. Their interaction defines selective pressure. Even microbial communities show genetic divergence across trench systems. Horizontal gene transfer occurs, but geographic separation still influences lineage development. As exploration continues, more examples of trench-specific evolution will likely emerge. Each discovery adds detail to understanding how life responds to extreme isolation. And as attention gradually moves toward the possibility of undiscovered species still waiting in unexplored trench regions, the role of isolation remains central, shaping biological diversity quietly over immense timescales beneath the ocean's surface, where environmental stability, pressure, darkness, and limited migration opportunities combine to produce evolutionary outcomes distinct from those observed in more interconnected parts of the planet. As attention shifts from the organisms themselves to the process of finding them in the first place, the question naturally arises: how do scientists even discover creatures living nearly 11 kilometers beneath the ocean surface? The Mariana Trench is not simply deep, it is remote, dark, cold, and under crushing pressure. Human divers cannot reach it directly. Even advanced equipment struggles under those conditions. And yet, over the past several decades, researchers have steadily built methods that allow glimpses into this otherwise inaccessible environment. The first challenge is simply getting there. Research vessels travel to the trench region equipped with specialized deployment systems. These ships carry remotely operated vehicles, autonomous landers, deep-sea cameras, sonar mapping tools, and occasionally crude submersibles. Each piece of equipment must tolerate extreme pressure, corrosion from seawater, and long descent times. Sonar mapping usually comes first. Ships emit sound pulses downward. When the sound reflects off the seafloor and returns, the time delay reveals depth. Multi-beam sonar systems send out many pulses simultaneously, producing detailed maps of seafloor topography. Before cameras descend, scientists want to know the terrain. Mapping identifies slopes, sediment plains, ridges, and potential vent sites worth investigating. Once a suitable area is selected, unmanned landers are often deployed. These are weighted frames carrying cameras, lights, bait containers, sensors, and sometimes sample collectors. They descend freely through the water column, a journey that can take several hours. Upon reaching the seafloor, they begin recording. Bait plays a surprisingly important role. In environments where food is scarce, scent travels far. Small pieces of fish or other organic material placed in bait canisters attract scavengers such as amphipods, snailfish, and other deep organisms. Cameras record arrivals, feeding behavior and interactions. This method reveals species that might otherwise remain unseen. Lighting requires careful balance. Powerful lights allow cameras to capture clear images, but sudden illumination can alter animal behavior. Some researchers use dimmer or red lighting less visible to deep-sea species. Advances in low-light camera technology have reduced the need for intense artificial illumination. Remotely operated vehicles, often called ROVs, provide greater control. Unlike free-falling landers, ROVs remain tethered to the ship via fibre optic cables. Operators on the surface control movement in real time. Cameras transmit live video. Robotic arms collect specimens, manipulate objects, and position sensors precisely. However, the tether length required for trench depths creates engineering challenges, and operations must account for cable weight and drag. Autonomous underwater vehicles represent another approach. These vehicles operate without tethers, following pre-programmed routes. They can map terrain, measure chemical gradients, and collect environmental DNA samples. Data retrieval occurs when the vehicle returns to the surface or transmits information acoustically. Crude submersibles provide direct human observation, though they are rare due to cost and risk. The bathyscaphe Trieste made the first descent to Challenger Deep in 1960. Later descents by advanced submersibles demonstrated improved materials, navigation systems, and life support. Inside these pressure-resistant spheres, observers view the deep environment through thick acrylic or specialized glass viewports. Specimen collection presents unique challenges. Bringing animals to the surface without damage requires maintaining pressure conditions as closely as possible. Pressure-retaining sample containers help preserve tissues. Without them, decompression can distort anatomy or affect biochemical processes. Environmental DNA sampling has become increasingly valuable. Water or sediment samples contain genetic fragments shed by organisms. Sequencing this DNA allows identification of species even if they are not directly observed. This technique reveals hidden biodiversity and helps map species distribution. Sediment cores provide additional information. Long tubes inserted into the seafloor capture layered sediment samples. These cores contain microorganisms, chemical signatures, and sometimes small invertebrates. Analyzing them reveals ecological history, nutrient cycles, and community composition. Chemical sensors mounted on landers or ROVs measure oxygen levels, temperature, salinity, pH, methane concentration, hydrogen sulfide levels, and other parameters. These data help explain why certain species appear in specific locations. Acoustic monitoring sometimes detects biological sounds. Although many deep-sea organisms do not produce loud noises, subtle vibrations can indicate activity. Hydrophones record ambient soundscapes, offering indirect evidence of life. Long-duration observatories represent a newer approach. Instruments left on the seafloor for months or years collect continuous data. These observatories monitor environmental stability, species arrival patterns, and long-term ecological changes. Technological innovation drives discovery. Pressure-resistant materials, advanced robotics, high-sensitivity cameras, and improved navigation systems all expand exploration capability. Each technological improvement allows access to slightly deeper or more challenging environments. Logistics remain complex. Deep-sea expeditions require significant funding, interdisciplinary teams, and careful planning. Weather conditions, equipment reliability, and operational safety all influence mission success. Data interpretation continues long after expeditions end. Video footage requires careful review. Genetic sequences need analysis. Chemical data must be correlated with biological observations. Collaboration among biologists, geologists, chemists, and engineers is essential. Historical context also matters. Early deep-sea exploration relied on dredging, which often damaged specimens and provided limited behavioral information. Modern imaging allows observation of animals in their natural posture and environment, improving ecological understanding. Public interest sometimes accelerates research funding. Documentaries, educational outreach, and high-profile expeditions raise awareness about deep-sea ecosystems. Increased awareness can support continued scientific investment. There are also ethical considerations. Minimizing disturbance to fragile ecosystems is important. Sampling strategies aim to balance scientific value with environmental protection. Many expeditions follow strict protocols to reduce impact. Satellite communication enables real-time collaboration. Scientists on shore can observe live ROV feeds, providing input during dives. This expands expertise available during exploration. Machine learning now assists with image analysis. Automated systems help identify species in large video datasets, accelerating discovery and reducing manual workload. Unexpected findings remain common. New species appear regularly. Known species exhibit previously undocumented behavior. Each expedition carries potential for surprise. Climate monitoring increasingly incorporates deep-sea data. Understanding carbon sequestration, oxygen distribution, and ecosystem resilience requires knowledge of trench environments. Education and training programs prepare new researchers for deep-sea work. Interdisciplinary expertise becomes increasingly valuable as exploration techniques diversify. International cooperation also plays a role. Ocean trenches span national boundaries and international waters. Collaborative research ensures broader data sharing and resource efficiency. Even with current technology, much of the trench remains unexplored. Mapping resolution varies. Biological surveys cover limited areas. The potential for undiscovered species remains high. And as exploration continues toward regions not yet surveyed and species not yet documented, the process itself remains an ongoing combination of engineering innovation, scientific curiosity, careful observation, and gradual technological refinement, allowing scientists to reveal creatures that exist far beyond direct human reach beneath the deepest parts of the ocean, where pressure, darkness, and distance make discovery possible only through persistent and carefully designed exploration methods. Even after decades of technological advancement, mapping improvements, and carefully planned expeditions, one fact remains consistent about the Mariana Trench. We have only seen a fraction of what lives there. Every time a lander descends or an ROV glides across the seafloor, it surveys only a tiny portion of an immense, complex landscape. The trench stretches for thousands of kilometers. Its slopes, ridges, sediment basins, and fault lines create countless microhabitats. And in many of those places, no camera has ever passed. When scientists analyze environmental DNA samples collected from trench water and sediment, they often find genetic sequences that do not match any known organism. These fragments suggest the presence of species never formally described. In some cases, researchers can determine that the DNA belongs to a new bacterial lineage. In others, it may represent an unknown invertebrate, or even a vertebrate species that has never been directly observed. The scale of potential undiscovered biodiversity becomes clearer when considering sampling limitations. A single sediment core may represent just a few square centimetres of the sea floor. A camera deployment might cover an area the size of a small room. Compared to the vast area of the trench, this coverage is minimal. Large regions remain unsampled. Microbial diversity likely represents the greatest unknown. Deep-sea sediments contain dense microbial communities. Many of these microbes resist cultivation in laboratory conditions. Without cultivation, traditional identification methods fail. Genetic sequencing has revealed surprising diversity, but even this approach captures only a portion of existing life. Invertebrates also contribute to the unseen majority. Tiny worms, crustaceans, mollusks, and other organisms inhabit sediment layers and crevices in trench walls. Many measure only millimetres in length. They rarely appear on wide-angle camera footage. Discovering them requires targeted sampling and microscopic examination. Even larger organisms can remain unnoticed. Some species may occupy niches away from commonly sampled areas. For example, steep trench slopes, vertical rock faces, and deep fissures receive less attention than flat sediment plains. These areas may host species adapted specifically to those microenvironments. Seasonal patterns at the surface could also influence deep populations in subtle ways. Variations in plankton productivity alter marine snow input. Organisms adapted to these cycles may display reproductive timing or behavioural shifts not yet documented because continuous monitoring is rare. Isolation between trenches increases the likelihood of endemic species. The Mariana Trench is not the only deep ocean trench on Earth. Others exist in different ocean basins. Genetic separation between these trenches may lead to species unique to each location. Even within a single trench, geographic separation between segments may promote divergence. Technological limitations further contribute to unseen biodiversity. Cameras rely on artificial light. Some organisms may avoid illuminated areas. Others may inhabit regions too narrow or unstable for safe equipment deployment. Delicate species might be destroyed during sample retrieval. Pressure-retaining containers help preserve specimens, but capacity remains limited. Scientists must choose carefully which samples to bring back. Many organisms observed on video are never collected physically, leaving formal description incomplete. Environmental DNA analysis expands discovery potential, but introduces interpretation challenges. DNA fragments degrade over time. Identifying whether sequences represent living organisms or remnants of long-dead individuals requires careful analysis. Long-duration observatories may reveal species that appear only occasionally. Some trench organisms may migrate vertically within pressure-tolerant ranges or respond to rare food falls. Short missions might miss these intermittent visitors. The possibility of unknown vertebrates remains particularly intriguing. While current evidence suggests vertebrate fish approach depth limits near the deepest trench zones, subtle variations in physiology might allow undiscovered species to exist slightly deeper or in isolated pockets. New vent systems or chemical seep zones may harbour unique communities not yet sampled. Hydrothermal activity varies over time. Previously inactive sites can become active, supporting new populations. These transient environments increase species turnover. Climate-driven changes may also influence deep biodiversity. Shifts in ocean circulation, oxygen levels, or carbon flux could gradually alter trench ecosystems. Some species may emerge or disappear without ever being documented. Machine learning applied to video footage has already identified organisms previously overlooked in archival recordings. Subtle shapes once dismissed as debris have been recognized as living creatures upon reanalysis with improved software. There is also the human factor. Scientific understanding evolves as expertise expands. A specimen once classified broadly may later be divided into multiple distinct species after detailed genetic analysis. Taxonomic work remains time-consuming. Formal description requires careful morphological examination, genetic sequencing, and comparison with known species. Many collected specimens await full analysis in research institutions worldwide. Funding and expedition frequency limit coverage. Deep-sea research is expensive. Each mission requires substantial logistical support. As a result, sampling remains intermittent rather than continuous. Despite these constraints, discoveries continue steadily. New amphipod species, novel bacterial lineages, previously unrecorded snailfish variants, and unexpected invertebrates have been documented in recent years. Each addition underscores how incomplete our inventory remains. Unseen species also represent potential biochemical diversity. Unique enzymes, metabolic pathways, and symbiotic relationships may exist undiscovered. These biological innovations could have applications in medicine, biotechnology, and environmental science. Public perception often imagines the deep sea as barren. In reality, it is underexplored rather than empty. The absence of light and direct human presence contributes to the illusion of emptiness. The trench functions as both habitat and evolutionary archive. Species evolving in isolation may carry genetic adaptations not found elsewhere. Protecting such environments preserves biodiversity we have not yet fully identified. International collaboration increases discovery potential. Shared data, open access genetic databases, and cooperative expeditions broaden research reach. Educational outreach also plays a role. Inspiring interest in deep-sea science encourages future researchers to pursue unanswered questions about unseen life. As exploration technology improves, including quieter propulsion systems, better imaging sensors, and longer-lasting autonomous vehicles, access will expand. Areas currently unreachable may become accessible. The possibility that entirely new branches of life remain hidden is unlikely, but not impossible. At minimum, numerous species within known groups almost certainly await formal recognition. Every descent into the trench carries both confirmation and uncertainty. Confirmed species reappear. Known ecosystems behave as expected. Yet occasional unfamiliar shapes remind researchers that the trench still holds surprises, and as attention continues shifting toward broader questions about what deep-sea life reveals about planetary habitability and long-term ecosystem stability, the idea that unseen species remain below the limits of current exploration remains both scientifically plausible and quietly compelling, suggesting that beneath the weight of the ocean, in regions cameras have not yet reached, evolution may still be producing forms and adaptations we have not yet imagined, waiting for future expeditions to reveal them gradually through careful observation and persistent inquiry. As exploration of the Mariana Trench continues to reveal organisms thriving under pressure, darkness, chemical exposure, and limited oxygen, scientists have begun looking beyond Earth when interpreting these discoveries. The conditions in deep trenches share similarities with environments believed to exist elsewhere in the solar system. That connection has shifted deep-sea biology from a purely oceanographic field into something closely linked with planetary science and astrobiology. For much of the 20th century, assumptions about extraterrestrial life centered on Earth-like conditions. Liquid water, sunlight, moderate temperature, and oxygen were considered essential. The discovery of thriving ecosystems in the deep ocean, completely independent of sunlight and capable of tolerating extreme pressure and chemical exposure, forced a reassessment. If life can adapt here, similar strategies might operate elsewhere under comparable conditions. One of the strongest comparisons involves icy moons such as Europa, orbiting Jupiter, and Enceladus, orbiting Saturn. Evidence suggests both possess subsurface oceans beneath thick ice crusts. These oceans likely receive little or no sunlight. However, Gravitational interactions with their parent planets generate tidal heating. This heating may produce hydrothermal activity on their ocean floors, similar in principle to Earth's deep-sea vents. On Earth, hydrothermal vent ecosystems rely on chemosynthesis rather than photosynthesis. Bacteria oxidize chemicals released from Earth's crust, creating organic matter that supports complex communities. If similar chemical gradients exist beneath extraterrestrial ice, microbial ecosystems could potentially arise there as well. Pressure in subsurface oceans on icy moons may equal or exceed that found in Earth's deepest trenches. Organisms in the Mariana Trench demonstrate that cellular chemistry can adapt to extreme compression. Stabilizing molecules, flexible membranes, and pressure-tolerant enzymes allow metabolism to continue. These biochemical strategies provide models for what extraterrestrial life might require. Temperature presents another point of comparison. Deep ocean water remains near freezing, similar to expected conditions in extraterrestrial subsurface oceans. Earth organisms show that life does not require warmth in the conventional sense, only stable conditions that allow biochemical reactions to proceed. Chemical energy availability is central. Methane, hydrogen sulfide, and other reduced compounds serve as energy sources for Earth microbes. Observations of plumes erupting from Enceladus suggest the presence of water vapor mixed with organic molecules and possibly hydrothermal minerals. This has led scientists to propose that chemosynthetic ecosystems could exist there. The absence of sunlight in these extraterrestrial environments parallels trench conditions closely. Photosynthesis becomes irrelevant. Energy cycles would depend entirely on chemistry, just as in deep-sea vent systems. Oxygen availability differs. Earth's deep ocean ultimately connects to an oxygen-rich atmosphere. Extraterrestrial oceans may lack comparable oxygen sources. However, some Earth microbes operate anaerobically using sulfate, nitrate, or other compounds instead of oxygen. Studying these pathways helps scientists imagine alternative metabolic systems. Isolation provides another parallel. Subsurface oceans beneath thick ice shells may remain isolated for millions or billions of years. Trench ecosystems, while not completely isolated, experience reduced gene flow compared to surface ecosystems. Evolutionary adaptation under isolation conditions on Earth offers clues about how extraterrestrial life might diversify. Microbial diversity in trench sediments suggests that life can persist in extremely stable, low-energy environments for long periods. Such persistence would be essential for extraterrestrial ecosystems where energy input might be limited. Planetary exploration missions increasingly incorporate instruments designed to detect chemical signatures associated with life. Knowledge gained from deep-sea microbial metabolism informs instrument calibration. Scientists look for isotopic ratios, organic molecules, and chemical disequilibria similar to those observed in Earth's extreme environments. Mars provides a different but related example. Evidence indicates that ancient Mars possessed liquid water. Some researchers speculate that subsurface brines may still exist. Earth microbes capable of surviving in saline, high-pressure, low-temperature environments provide analogues for potential Martian life. Studying deep-sea organisms also informs planetary protection protocols. Preventing contamination of extraterrestrial environments with Earth microbes requires understanding which organisms can survive extreme conditions. Trench microbes demonstrate remarkable resilience, influencing sterilization standards for spacecraft. Another insight involves evolutionary flexibility. The range of adaptations seen in trench organisms shows that life does not follow a single template. Multiple biochemical solutions exist for coping with pressure, darkness, and chemical stress. This diversity suggests extraterrestrial life, if present, might not resemble familiar surface organisms. Bioluminescence provides an interesting example. In permanent darkness, Earth organisms generate their own light for communication, predation, or camouflage. While extraterrestrial oceans may lack complex animals, Similar signalling mechanisms cannot be ruled out if multicellular life exists. Energy efficiency remains a recurring theme. Deep-sea organisms often exhibit slow metabolism and long lifespans. Extraterrestrial ecosystems with limited energy input might show similar traits. Detecting slow-growing microbial communities would require sensitive instruments and long observation periods. Geochemical cycles in the trench illustrate how life interacts with planetary processes. Microbes influence carbon sequestration, sulfur cycling, and mineral deposition. Understanding these interactions helps scientists interpret extraterrestrial geochemical data. There is also caution against overinterpretation. Similar environmental conditions do not guarantee similar biological outcomes. Earth life evolved under specific historical circumstances. Extraterrestrial environments may differ in subtle but important ways. Nevertheless, deep-sea research expands the definition of habitability. Environments once dismissed as too extreme now appear plausible hosts for life. This shift influences mission planning, target selection, and instrument design in planetary exploration. Public interest in astrobiology often increases support for deep-sea research. Discoveries in one field inform the other. Collaboration between oceanographers, microbiologists, planetary scientists, and chemists continues to grow. Technological advances developed for deep ocean exploration, such as pressure-resistant sensors and autonomous vehicles, inform planetary probe design. Conversely, space mission technologies sometimes improve oceanographic equipment. Ethical considerations also arise. Protecting extreme Earth environments becomes more important when they serve as analogues for extraterrestrial habitats. Preserving their integrity ensures accurate scientific interpretation. Education benefits as well. Connecting deep-sea biology with the search for extraterrestrial life captures public imagination and encourages interdisciplinary study. Despite similarities, Earth's deep ocean remains more accessible than extraterrestrial oceans. Continued exploration here offers immediate opportunities for discovery and hypothesis testing. And as exploration moves gradually toward understanding what these extreme environments reveal about planetary processes, evolutionary resilience, and the limits of biological adaptation, the organisms thriving in the Mariana Trench continue to demonstrate that life can persist under conditions once considered incompatible with complexity, providing a practical reference point for scientists considering where life beyond Earth might exist and how it might function in environments defined not by sunlight and surface warmth, but by pressure, chemistry, and long-term environmental stability. As attention settles on the strange and often unfamiliar creatures of the Mariana Trench, it becomes easy to treat them as curiosities, distant biological oddities that exist far removed from everyday human life. Soft-bodied fish drifting through darkness, crustaceans larger than expected, bacteria metabolizing chemicals most animals cannot tolerate. From the surface perspective, these organisms might appear disconnected from practical concerns. Yet the reality is more complex. The life forms inhabiting the deepest ocean play roles that extend beyond their immediate environment, influencing global systems, scientific understanding, and even future technological development. One of the most significant reasons these creatures matter involves global nutrient cycles. The deep ocean acts as a major reservoir for carbon. Organic material sinking from surface ecosystems eventually reaches trench environments, where microbes and scavengers process it. Some carbon becomes buried in sediments, effectively removed from the atmosphere for long timescales. This sequestration influences Earth's climate system. Microbial activity, invertebrate feeding, and sediment dynamics all contribute to determining how much carbon remains stored versus how much re-enters ocean circulation. The biological carbon pump, as scientists often describe it, relies heavily on deep-sea ecosystems. Without organisms breaking down organic material and redistributing nutrients, surface productivity patterns would shift. Even though trench organisms are physically distant from human society, their cumulative effect contributes indirectly to atmospheric composition and climate regulation. Biodiversity represents another critical factor. The Mariana Trench hosts species found nowhere else on Earth. Preserving biodiversity matters not only for ethical or ecological reasons, but also for scientific discovery. Many deep-sea organisms produce unique biochemical compounds. Some enzymes remain stable under extreme pressure or cold temperatures. These properties attract interest in biotechnology, medicine, and industrial chemistry. For example, enzymes derived from deep-sea microbes may function effectively in high-pressure industrial reactors. Others show potential in pharmaceutical development, including antimicrobial compounds. The full range of biochemical capabilities remains largely unexplored because so many species remain undocumented. Studying trench organisms also advances basic biological understanding. Life in extreme environments challenges assumptions about physiology, metabolism, and structural adaptation. Discoveries such as pressure-stabilizing molecules, flexible cellular membranes, and hypoxia tolerance expand knowledge applicable to medicine and biotechnology. Medical research sometimes draws inspiration from extreme environment biology. Understanding how deep-sea organisms protect proteins from pressure-induced damage may inform treatments for certain human conditions involving protein misfolding. Hypoxia-tolerant studies contribute to knowledge relevant to cardiovascular health, stroke treatment, and critical care medicine. Environmental monitoring benefits as well. Deep-sea organisms serve as indicators of global change. Pollutants such as persistent organic chemicals and microplastics have been detected even in trench amphipods. Tracking these contaminants helps scientists understand how human activity spreads through ocean systems. Climate change research increasingly incorporates deep ocean data. Temperature trends, oxygen distribution, and carbon sequestration processes all involve trench environments. Without understanding these regions, global climate models remain incomplete. There is also the question of ecological resilience. Deep ecosystems operate under stable conditions but may respond slowly to disturbance. Monitoring trench communities provides early warning of long-term environmental shifts that might otherwise remain unnoticed. Technological innovation often follows scientific discovery. Pressure-resistant materials, advanced sensors, autonomous vehicles, and imaging systems developed for deep-sea exploration have applications beyond oceanography. Engineering solutions designed for trench research sometimes translate into industrial, environmental, or medical technologies. Educational value should not be overlooked. The existence of life under such extreme conditions expands public understanding of biology and environmental science. It encourages curiosity about Earth's less visible systems and promotes interest in scientific careers. There is also a philosophical dimension. Recognizing that complex ecosystems exist in places humans rarely see can influence attitudes toward environmental stewardship. Awareness of hidden biodiversity encourages broader perspectives on conservation. Economic considerations occasionally arise. Deep-sea mining proposals target mineral deposits in some ocean regions. Understanding biological communities before industrial activity begins is essential for responsible decision-making. Trench ecosystems, though less targeted than some abyssal plains, still require protection from unintended impacts. Legal frameworks governing deep-sea environments continue evolving. International cooperation helps balance scientific exploration, conservation, and potential resource use. Knowledge of trench biodiversity informs policy discussions. Cultural influence also plays a role. Documentaries, educational programs, and scientific publications about deep-sea life shape public perception of the ocean. Increased awareness can support conservation initiatives and scientific funding. From an evolutionary perspective, trench organisms preserve information about adaptation under extreme conditions. Studying them helps reconstruct evolutionary pathways and understand how life responds to environmental stress over long timescales. Astrobiology benefits directly. As discussed earlier, deep-sea ecosystems serve as analogues for potential extraterrestrial habitats. Understanding these systems improves interpretation of planetary exploration data. The interconnectedness of ocean systems means trench processes do not remain isolated. Currents circulate nutrients, chemicals, and organisms throughout global oceans. Changes in one region can influence others gradually. Ethical considerations arise regarding exploration itself. Minimizing disturbance to fragile ecosystems ensures that scientific inquiry does not inadvertently damage what it seeks to understand. Responsible research practices remain essential. Public engagement often increases when deep-sea discoveries capture imagination. Interest in unusual creatures can serve as an entry point for broader environmental education. Long-term monitoring of trench ecosystems contributes to baseline datasets. Without such baselines, detecting change becomes difficult. Establishing current conditions allows future comparison. Technological miniaturization may enable more frequent, less invasive observation in coming decades. Continued innovation will likely expand access to deep environments while reducing impact. Scientific collaboration across disciplines remains crucial. Oceanography, microbiology, geology, chemistry, engineering and planetary science all intersect in trench research. Even aesthetic appreciation has value. Observing unfamiliar life forms broadens perspective on biological diversity. This perspective often encourages conservation-minded attitudes. The notion that these creatures exist far from daily human experience does not diminish their importance. Global systems link surface and deep environments continuously. As research progresses, new connections between trench ecosystems and broader Earth systems continue to emerge. Each discovery adds nuance to understanding planetary function. And as attention gradually moves toward final reflections on the deep ocean as a whole, the creatures inhabiting the Mariana Trench remain significant, not because they are unusual alone, but because their existence informs climate science, biotechnology, medicine, planetary exploration, environmental policy, and fundamental biological knowledge, demonstrating that even in the most remote parts of the planet, life contributes meaningfully to systems that ultimately affect the surface world where human societies operate. As the exploration gradually settles and the final images of the trench begin to fade from view, it becomes natural to step back for a moment and consider what all of this really means. Not just the strange fish, the glowing organisms, the chemical-eating bacteria, or the immense pressure, but the simple fact that an entire world exists below us most of the time unnoticed, operating continuously whether we pay attention to it or not. The deep ocean does not announce itself loudly. It does not change quickly enough for casual observation. It simply continues, stable, quiet, and persistent. Yet within that stability, life has adapted in ways that stretch expectations. Creatures without sunlight, organisms thriving under pressures that would crush most familiar animals, ecosystems fueled by chemistry instead of light. These realities expand how we understand what is possible for life on Earth. There is something grounding about recognizing how much of our own planet remains only partially explored. Human technology has reached space, crossed continents, and mapped much of the surface world, but the deep ocean still holds unanswered questions. Entire species remain undocumented. Ecological interactions continue beyond direct observation. The trench, in particular, represents one of the clearest reminders that discovery is ongoing. At the same time, deep-sea research has already reshaped multiple areas of science. Biology, geology, climate science, chemistry, and planetary exploration all draw insights from these environments. Knowledge gained from trench organisms influences understanding of protein stability, metabolic flexibility, global carbon cycling, and even the possibility of life beyond Earth. The connection between deep ocean ecosystems and surface conditions often surprises people. What happens thousands of meters below influences nutrient cycles, carbon storage, and chemical balances that ultimately affect atmospheric composition. The relationship may be indirect, but it is continuous. Ocean systems function as integrated wholes rather than isolated layers. There is also a reminder about scale. Human experience tends to focus on environments within direct reach. Cities, landscapes, weather patterns. The trench operates on spatial and temporal scales that differ significantly. Evolution there unfolds slowly. Environmental changes accumulate gradually. Patience becomes a defining characteristic of both the ecosystem and the science studying it. Technology continues improving access. Autonomous vehicles remain underwater longer. Sensors become more sensitive. Imaging systems capture clearer detail with less disturbance. Each advancement increases the likelihood of new discoveries. Yet even with improved tools, the trench retains an element of quiet mystery, simply because of its vastness. Public interest in deep-sea life has grown steadily. Documentaries, educational outreach, and scientific publications bring images of these environments to wider audiences. That awareness helps support conservation discussions and research funding. Increased understanding tends to encourage thoughtful stewardship. Environmental considerations remain important. Pollution has reached even the deepest ocean. Microplastics and persistent chemicals appear in trench sediments and organisms. These findings emphasize how interconnected planetary systems are. Actions at the surface eventually influence remote environments. Climate change introduces additional variables. Ocean temperature, oxygen distribution, and carbon cycling patterns may shift gradually. Monitoring deep ecosystems contributes to understanding those changes over long time scales. Scientific collaboration continues expanding. International teams share data, coordinate expeditions, and develop joint research initiatives. Ocean trenches often lie beyond national boundaries, encouraging cooperative approaches. Education benefits as well. Deep-sea research illustrates scientific method in action: hypothesis, exploration, observation, revision. It shows how knowledge evolves as new data appear. There is also personal perspective. Recognizing that life thrives in places once thought uninhabitable can influence how people think about resilience, adaptation, and environmental limits. It challenges assumptions gently, through evidence rather than speculation. For some, deep ocean exploration sparks curiosity about broader planetary processes. Plate tectonics, ocean circulation, chemical cycles, evolutionary biology. The trench becomes an entry point for wider scientific interest. For others, it simply provides a sense of scale and continuity. The planet functions through systems far older than human history. Those systems continue whether observed or not. Future exploration will likely reveal additional species, new metabolic pathways, and perhaps entirely unexpected ecological relationships. Each expedition adds incremental knowledge rather than sudden complete understanding. Preserving these environments while studying them remains a priority. Responsible exploration minimizes disturbance while maximizing information gained. There is also the practical benefit of technological innovation driven by deep-sea research. Materials science, robotics, sensor development, and data analysis techniques often advance through the demands of exploring extreme environments. Astrobiology remains closely connected. Understanding how life adapts to pressure, darkness, and chemical energy informs the search for life elsewhere. The trench offers a terrestrial reference point for conditions once considered improbable for biology. Despite all these scientific implications, there remains a simpler element that does not require technical explanation. Observing a soft-bodied fish drifting calmly under immense pressure, or a faint bioluminescent glow appearing briefly in total darkness can produce a quiet sense of perspective. It reminds us that life does not conform to a single familiar template. The deep ocean does not ask for attention, but when attention turns toward it, the response is often a mixture of curiosity, respect, and thoughtful reflection. These environments have existed for millions of years. They will likely continue long after current technologies change again. And as the images of the trench gradually recede and the broader ocean returns to its usual quiet background presence, the understanding remains that beneath the surface, far below everyday awareness, a complex and persistent world continues steadily, shaped by pressure, chemistry, time, and adaptation, offering ongoing opportunities for discovery whenever curiosity and careful exploration bring it briefly into view again.